Okay, everybody, we're going to go ahead and get rolling this morning. Good morning, everyone. Hope you had a great first day of the conference. Hope you're enjoying the conference. Our first speaker this morning is Dr. Stephen Benner.
All right.
Take it away, sir.
So I'm circulating through the audience right now a small rock, which is a clam, which has vivianite, the iron phosphate mineral that they discussed a couple weeks ago at the press conference. Today I'm just going to go through these propositions that are, I think, more likely true than not. I mention that because this is what we call the preponderance of evidence standard. So if you go to a court of law and they accuse you of a criminal trial where you can be put in jail, they have to prove your guilt beyond a reasonable doubt. The lawyer might try to say he's going to prove your guilt beyond a shadow of a doubt. But in a civil trial, it's just a preponderance of evidence just means that these propositions are more likely than not. And of course, that means you get a discussion started. So the propositions are relatively direct that life emerged on Mars about 4.3 billion years ago. Second, macroscopic life perhaps even lived on Mars about 3.2 to 3.8 billion years ago. Life survived until today, adapting to Mars as the climate cooled and dried, as you heard about yesterday. So Viking in 1976, the proposition detected that surviving life. That we have tools now to conclusively identify Martian life as life if we come across it. And you can do your own Martian astrobiology. You can participate this. So the price of entry is about $15,000 per kilogram. This is some discussion that Jan Spacek will also have early this afternoon in what is called the IMPRESS, which is the International Mars Penetrated Ride-Sharing System architecture to get small, even garage scientists designed experiments on Mars. So let's go through and have a quick look at them. So what's passing around right now is the vivianite. And you may have seen this paper or maybe a press conference. I did an Event Horizon podcast on this a couple of days ago by Joel Horowitz, who have claimed that they are seeing these particular structures on the surface. Now, you're looking for these dark green objects. What I'm passing around here is exactly the same material, which is vivianite. This is actually coming from the Kerch Peninsula in a 20-million-year-old clam who met his unfortunate death, of course, in that area. That's actually the peninsula in Crimea that juts towards the bridge that connects Crimea to Russia, which is well in the news these days. But this guy shows you what happens and why people think that this vivianite, these dark green spots, are themselves signatures of life on Mars at the age of these particular sediments, which are not well dated. They're somewhere between 3.2 and 3.8 billion years. So the idea here is that the organic materials in that clam that is circulating around you nucleate the growth of iron phosphate. Now, iron phosphate is relatively insoluble, but is by no means the most insoluble of all salts that are possible. So calcium phosphate is much less soluble than iron phosphate. And as we'll see on the next slide, there is calcium in this environment. In fact, what I love to say is that this is the geologist thing. Every rock has its story. So if I hand you a rock, you're supposed to be able to tell me its story. And the rocks here, these are the leopard spots. But there's a calcium sulfate vein that was infused later. There's a lot of calcium around. This little black crystal is a crystal of olivine, which is a magnesium iron silicate, which comes volcanic and washed there. This is what we call in geology detrital. It is delivered there by flowing water. And so this material has formed iron phosphate when there's calcium around and could have formed calcium phosphate, less soluble by 4 or 5 orders of magnitude. So the question is, why is there iron phosphate, for example, in that particular sample that's circulating around you? Because the shell is calcium carbonate. And the answer is because organic material has a special property is that it nucleates the growth of iron phosphate crystals. So if you have organic material and you have iron phosphate, iron in the medium and phosphorus in the medium, you nucleate the growth of iron phosphate. Now, keep in mind, you concentrate phosphate and iron from your environment. You go out there and sit yourself in the desert among rocks. What's concentrated in the rocks relative to you is silica. And you don't have much silica. But you go ahead and concentrate the iron and phosphate, and the clam did so as well. So when that clam died, he had already lots and lots of iron phosphate to form these crystals. Iron phosphate is not the preferred thing to do in any particular environment, except when you're in a life form which has concentrated the iron and the phosphate from the surrounding rocks and minerals. And you have the organic material from the decaying clam nucleating the growth of these and fixing that iron phosphate in a crystal form before it can get access to the calcium. So this is the reason why this looks like a biosignature to us. And again, it's not proven. It's more likely than not. But one of the key questions that we have to worry about is to what extent this material has ever been below ground. You're looking at detrital rocks that are pebbles like the olivine that are being thrown into this rock, which would apply a flowing water on a surface. So if this has never been particularly buried, there's really no other way to get that iron phosphate. And so that's why this is more likely than not your first proposition is true. So the second proposition is that life emerged on Mars 4.3 billion years. Now you could say, hey, if I take that as a sign of life on Mars at 3.5 billion years, it has to have emerged. Life must have emerged before then. But there's actually reasons in which we can go back and draw chemistry that will do this. I'm actually showing you here a picture of RNA that's a genetic molecule. It's actually what COVID uses to have kids, right? It's a genetic material. You do mostly DNA for your propagation, but RNA is an alternative. And it's more importantly, it's believed that RNA was the first alternative. That is, that RNA emerged by interactions between chemicals and rocks on a primordial Earth. And what you're seeing here is a laboratory example. Keep in mind that RNA is a long molecule. It has repeating negative charges in the backbone. It is therefore what we call a polysalt or a polyelectrolyte. So you're actually able to move RNA in an electric field. And this experiment takes triphosphate, nucleoside triphosphates. It dumps them on basalt, which is an igneous rock. You can see lots of it in Iceland and Hawaii. This is what was present a lot on early Earth. And the basalt catalyzes the formation of RNA, which when we put the stuff in on the top, the electric field pulls the material down through this gel. The smaller molecules travel faster than the larger molecules because these are able to make their way through the gel, which is sort of a crowded environment, faster. But what happens is after a couple of days, you start forming oligomeric RNA, which is the compound that would have provided the first genetic information management for the first forms of life, any place where you have these precursors, including the basalt. So where did this particular basalt come from? Well, just for the record, this particular salt, which Elisa Biondi and Su-Wei Peng did, came from Mars. Now, it didn't come to us directly. It didn't come from sample return, right? It came by having something hit Mars, eject the rocks, which traveled around the solar system for a while, and then landed on the Sahara Desert, where the camel herders know that there are some Western crazy people who will pay them good money for this rock. And so that's how this rock gets into our hands. But you are seeing on that particular gel the prebiotic synthesis of RNA on Martian basalt. Now, this is a more complicated story, which involves a lot of chemistry, which is not appropriate for this audience, but I thought I would put the slide up with a lot of chemistry just to tell you what's going on here. The key ingredient by this is borate, borax, which you sometimes use, or in the good old days, used in laundry detergents, which Ronald Reagan at one point, had 20 mule teams and a whole Western television series. The borate is very important because the borate is what allows you to get the triphosphates that you're using to make the RNA on Martian basalt. And that actually is coming from a chemical process where the ribose, the R in RNA, is coming from a borate-controlled maturation of carbohydrates that are being formed in an atmosphere that's made up of carbon dioxide and water and maybe some nitrogen. Now, guess what? The Martian atmosphere is made up of carbon dioxide and forms these molecules on the left. And more important, Curiosity rover found that Martian surface has a lot of borate. So what you're looking at here is what we love to call privileged chemistry. This is chemistry that could not have happened Mars has a large amount of borate on its surface. It's forming the molecules that are in red on the left-hand side of this. These molecules are being trapped by sulfur dioxide, which is coming out of Mars volcanism. All of this chemistry can operate on Mars, which is why we say that there's more likely than not that Martian life arose on Mars. Now, about the Martian life surviving until today. Now, remember, the Earth has also undergone dramatic changes in the environment, right? You and your kind, well, actually, cyanobacteria have put poison into the air, which is this dioxygen. You have adapted to it. Dioxygen is poisonous, very toxic to any organism that does not manage the oxidizing radical power of O2. But you've done it. And the argument is that Martian life did this again itself. And you got to keep in mind that this is Martian life, especially where the Viking sites went, but also where Perseverance rover goes. In fact, Mars sends its rovers to sites where the resources are especially scarce. This is a planetary protection idea. There are what they call special regions which have water and all sorts of goodies for life, but they're afraid that that would allow Earth life to survive in those environments if the spacecraft brought it. And so we stay away from the regions looking for things on Mars where life is most likely to exist today. But in any case, like any life form in a scarce resource environment, the Martian life would survive by being dormant. Okay, that's what happens in Antarctica or wherever you are in the Atacama Desert, wherever you have scarce resources. But then being prepared so that when you get food, water, some energy, something that needs it, you are able to instantly emerge when the resources become available. And if you actually ask yourself, if I'm sitting on Mars, what kind of lifestyle would I like to have to be prepared under these circumstances in a resource-scarce environment? The answer is what we love to call as a bacteria. It's a microbe similar in size to Earth bacteria. It's an autotroph, which means it is able to make its own stuff from carbon dioxide. It fixes carbon dioxide from the atmosphere. It is able, however, to respire. So once it's like a plant, once you fix the carbon dioxide by photosynthesis in carbohydrates, then you can take the carbohydrates as food and later burn them, eat them, use them to make more plant, this type of thing. You yourself are a heterotroph. You actually don't make your own organic material by fixing carbon dioxide. You have to eat plants, well, you eat animals who have eaten animals who have previously eaten plants. Plants and this autotrophy is at the bottom of your food chain. But I would have that also on Mars, something that's respiring. And it's respiring with oxygen that is stored. Remember, Martian atmosphere does not have much O2 needed to respire food. It's only 0.1%. So on Earth, if I'm a plant and I make carbohydrates during the day and I eject the oxygen into the atmosphere at night, I'm perfectly able to get that oxygen back from the atmosphere because it's all in the atmosphere. And since I'm a plant and I use sunlight to make the stuff, I'm on the surface, that's how I get the sunlight, but it's also how I can recover the oxygen. But the problem with the photosynthetic microbe on Mars is that if it throws its oxygen away into the atmosphere during the day as it's making organic material, it cannot necessarily recover that oxygen at night to be able to use the food that it has made during the day. So it's going to store the oxygen. And Barsoom is a bacterial autotroph respiring with stored oxygen for overnight metabolism. Happens to be, for those of you who are familiar with John Carter and the Princess of Mars, the name in which the Martians call their planet. Coincidence? I think not. In any case, so oxygen storage is the principal difference between a Martian and an Earth photosynthetic autotroph. On Earth, the plants can discard the O2 made during the day. They can recover the O2 at night from the air. A Martian really cannot. But now we go to the next proposition. Again, we're talking about, oh yes, this is my cartoon from Jake Fuller, who you are noticing, if you look closely, that the Martians are carrying around with them a little tank of oxygen to be able to imbibe and use the food that they have made or somebody else has made during the day. Cool. So the next proposition is that Viking in 1976 actually detected Barsoom life. Now, this is not what you have seen in your textbooks. And just yesterday, we finally got into Science magazine, and you can go look it up yourself at what's called an E-letter. The reason why Viking in 1976, the results were misinterpreted, the misinterpretations were then propagated in review articles, and then They were propagated in the reviews of the reviews, and those ended up in textbooks, and they all said that Mars didn't have life, was because in a science paper in 1976, 50 years ago, Klaus Wiemann decided that the organic molecules that he has seen in the Martian sample were contaminants brought from Earth. And of course, some of them were. Freon was seen on Mars, but freon had also been seen by the same instrument in flight out to Mars. So there's no question that freon is a contaminant. But Beeman also saw methyl chloride, methylene dichloride. These are now interpreted as the oxidation products of Martian organics in Martian soil containing Martian perchlorate, which is an oxidant that is able to oxidize organics when you heat it up to 500 degrees centigrade, which is what Klaus Wiemann's machine, what Viking in 1976 did to the soil samples. And they saw the methyl chloride, the methylene chloride, and we now understand that as proof of the presence of organics, not the proof of presence of the absence of organics. And so the problem was in the science paper, they made one lousy mistake. They put the methyl chloride and the methylene chloride in as terrestrial contaminants, even though these had not been seen on the flight out to Mars. And then the guy who reviewed the review was a man named Norm Horowitz, who referred to these as cleaning solvents. That's because Norm was a biologist and did not know the difference between ethylene chloride, which is what the dry cleaners use when you take your suit out to be dry cleaned, and methyl chloride. Methyl chloride is a gas that boils at minus 26 degrees centigrade. There's no way you're going to use that as a cleaning solvent, but there it is on Mars. And because that was moved, Horowitz got it wrong, and by the time you get to Jonathan Lunin's astrobiology textbook, he will tell you that no organics were seen on Mars. And by the way, he also gets the label release experiment wrong and the photosynthesis experiment wrong in the textbook. So there you go. So let's go see what the Martian Viking 1976 experiment did. Well, what they did was they took soil samples and they dumped them over in the left. They exposed them to radioactive carbon dioxide. They're looking for autotrophy. They're looking for photosynthesis. They're looking for the fixation of radioactive carbon dioxide, which they give it, and they're hoping that the Martian microbes living in the soil sample will fix that into organics, and they saw it. Okay? One for life detection. Another sample was run by Gil Levin and Patricia Stratt. It's a very famous experiment. They put food, radioactive food, onto the Martian soil sample, and the understanding of trying to find respiration. Heterotrophy, that is, they wanted to see carbon dioxide released from the food. Radioactive carbon dioxide, because the food was radioactive. If I feed you radioactive food, you exhale radioactive carbon dioxide. And they looked for the radioactive carbon dioxide, and they saw it. To life detection. The third experiment was a little bit more agnostic with respect to metabolism. They just reasoned that, well, if you have a soil with an active microbiome, it should be exchanging gases. We don't know what gases. So they look for hydrogen, and they look for nitrogen, and they look for ammonia, they look for methane, they look for water, they look for oxygen. And they saw exchange of carbon dioxide, and they saw release of the oxygen, which of course is the stored oxygen. That's the SO, stored oxygen for overnight metabolism, the SOOM of Barsoom. So they saw it, and they saw a respectable amount of that material. Now, it's a long story, which you can go to Science Magazine. Again, it's an e-letter attached to the original 1970. We tried to get them to retract the paper because they were Science is now into retracting papers. I think it's a good idea, but the bottom line is they said no. But you can go see now. A bunch of us, Dirk Schultz, McCooch, Jan Spacek, who's in the back here, a number of people put together a note on the paper which shows why the paper was wrong, and that's where all of this problem started. And now we have to work through 50 years of error as we gnaw back to try to correct this. Okay, so you don't know that Viking discovered Barsoom life on Barsoom because it is an immersive interpretation of gas chromatography, mass spectrometry, which I won't talk about anymore. But there's the e-letter, which you can go and dig up if you're interested. So that's the next proposition now, is that Martian life can be conclusively identified if we were to trip across it. Right. I mean, remember, we think that Martian life emerged. I got you RNA being formed on Martian basalt with substrates for that formation coming out of Martian available material. we see vivianite, and that sample that is being passed around is that clam growing iron phosphate vivianite crystals on the nucleation of organic material. Now, of course, the ones on Mars are smaller. Of course, the life is microbial, not macroscopic like that clam. But nevertheless, it's large enough to see, which is why I've teased you with this idea that macroscopic life might have been there. Life survived until today, adapting to the Martian environment. We can say what we think that life would have as its lifestyle. And then Viking, in fact, saw that lifestyle and all of its details. And so now we have to worry about finding it. So we've got to send a mission. Of course, that's a bit of a problem at this moment, because almost all the Mars, yeah, just pass that around. Make sure everybody gets a chance to see that and hold that in their hands so that they can actually see the actual vivianite crystals growing on organic material. So yeah, so regardless of origins, in water, we believe that information needs are supported by a biopolymer with a repeating backbone charge. It's negative or positive. RNA is negative. That's why we call it a polyelectrolyte. That's why it moves on that gel, as I showed you earlier. But we understand this because we have synthesized many alternatives, maybe about 400 now synthetic biology, which you'll hear a little bit more about in the next talk, have been made by chemists like me trying to find molecules other than DNA or RNA that are able to support the informational needs of evolution. So here's a quiz for you. If you want to find life on Mars, what might you look for? Well, the answer is sort of given to you, right? You're looking for a polymer with a repeating backbone charges that are built from size interchangeable building blocks, because regardless of its origins, evolution needs to be supported by a biopolymer with a repeating backbone charge, negative or positive, with size uniform building blocks. And you will look for this in Martian water, maybe in the ice permafrost, something. And so this is actually a device that is called the Agnostic Life Finder, or ALF. This is something which I'll show you a picture of Jan in a minute, and the device itself. You're looking at the flow of water, which contains particulates, which are the orange things, that are passing down the main channel. But you have membranes here and there, and you're operating between a negatively charged cathode and a positively charged anode. So if you put into this flowing water, instead of, in addition to particulate matter, you put in RNA from, say, Martian life, this is the green molecules. They're being attracted towards the positively charged thing. They can get through this membrane because the membrane has big holes. They can't get through that membrane because the membrane has smaller holes. The salts get through here. And if Martian natural history has been different than natural history on Earth, you might have positively charged genetic information molecules, and of course they're going to be attracted towards the cathode. And so again, they can make it through the first membrane because the first membrane has relatively large holes, but they can't make it through the second membrane. So what we're doing here is now therefore concentrating the informational biopolymers from the flow of Martian water, and you can concentrate them very especially. So you can now participate in the Mars astrobiology program, however, and this is the point. I'm hoping that this gentleman who is back there, who I should get him to, yeah, he'll be talking, I think, at 1, but there's an international Mars penetrator ride-sharing system, so don't listen to me. Right? Have an idea for something that you could do a little experiment on Mars, and then you have to get $15,000 by a bake sale or selling lemonade. And you can put this now into a shared ride system, and Jan is trying to organize that right now to get to Mars a bundle, maybe 1,000 of what are called penetrators. So they're going to come in. There's the delivery device. These are the individual penetrators. They're not going to have a soft landing, and that's because soft landings are expensive. But what you do have is a way of hardening the devices so that they're slowed down a little bit, and then they penetrate the source, maybe 30,000 to 50,000 gravitations of deceleration force, and you're supposed to be able to put an experiment in and into hardness so that your experiment survives the impact. And Jan has got lots of ideas on how to do the hardening so that it survives the impact. So there you go. This is actually, I believe, a penetrator. I think it was designed by Gil Levin. Gil Levin is the guy I mentioned earlier who did the label release experiment where he fed radioactive food to the surface and saw CO2 that was radioactive come out of the surface. That was one of the signs of respiration of life. But the summary of the talk is that if you set up your lemonade stand, collect $15,000 from local people who want to have their logo on the side of your experiment, you can go beyond, get yourself participating in the Mars exploration for $15,000. And the reason why we think that there is a likelihood of your being successful, again, it's not proof, it's preponderance of evidence. It's more likely than not that life emerged on Mars, that we have seen its traces from 3.5 billion years ago. It's survived today, adapting to Martian change. It was detected in 1976 by Viking. It can be identified once we encounter it. For a relatively small cost, you can go look for experiments yourself. With that, let me stop. Thank you for your attention. I'll be happy to answer any questions you have.
So the way you were talking about the Barsoom kind of idea where they're storing oxygen for later, and they're kind of waiting for resources until they appear and then they become active when they do. That sounds very similar to the bacterial life and microbial life in like the deep biosphere of Earth.
Yeah.
So do you think that we could learn more about the possible life on Mars by studying deep microbial life in the deep biosphere?
Yes, but you know what the most important thing in science is? What is the most important thing in science? Does anybody know? Money. Yes. So obviously, yeah, no, there's no question that we have learned a lot in 20 years. What happened in 1996 was that Dave McKay discovered what he thought were cell structures in a meteorite that came, sort of how I got my hands on a meteorite, sort of the same way, but this was collected in Antarctica. Bill Clinton, the president at the time, went allegedly to Dan Golden, who was running NASA at the time, and said, hey, is this life or isn't it? And Golden says, I don't know. And Clinton is reported to have responded, well, what the hell are we paying you for, right? You're the guy, when we get invaded by little green men, I'm going to come to you and ask, and you don't know. But Golden was a brilliant administrator, so he managed to get 20 years of a NASA astrobiology program and a lot of the work that was done to let us understand how life would survive in extreme environments was exactly the kind that you just suggested. So there's a huge literature on it, but it's incomplete. And the more we know about both Mars and exotic but resource-scarce environments on Earth, the more we need to learn to study.
I've got two questions which are diverse, so I'll just say them both quickly and then you can answer them. One is the early Earth, before we had an oxygen-rich atmosphere, the first life would be in the same boat as the Mars life is now. So can we find fossils or, for instance, as the young man said, look for survivors of the early Earth that have to store oxygen? Well, I guess the deep ones can't, but fossils that would indicate this form of life existed on the early Earth. And the other question is completely different, which is this. We currently have 2 operational rovers on Mars,
Yes.
and may have a third one launched in a couple of years from the Europeans. Is there anything that can be done with these rovers that could be, if you could direct these rovers as they are, to help confirm this theory?
Yeah, no, absolutely, both good questions. So in the first place, you're an organism that stores oxygen, right? You have hemoglobin and you store it to maybe 700 picomoles per milliliter of your blood. Now, you're not, that's not the primitive storage capacity, right? You're storing it because you're multicellular and you have a hard time getting, just by fluid mechanics, the stuff from outside inside. But the answer is we don't know, right? There's an enormous amount of organisms that live without oxygen at all. These are anaerobic things that live down deep in the ocean, but or in muck or in rocks, subsurface. So a lot of those have been studied, but as far as I know, none of them have been shown to store oxygen for long periods of time. But of course, they don't have oxygen in their life cycle in any case because they're removed from the atmosphere. So the short answer to your question is we don't have enough information about ancient forms of life on Earth, partly because the fossil record. I could have put up a slide of what we know about Earth 3.5 billion years ago, and it's a stromatolite that is, among other things, able to fix iron phosphate. But there's one site on Earth that has that, right? And so where our record has been And so that's one of the reasons why I think Mars is very exciting, because the record hasn't been subducted by loss by continental drift. So that's where we have to go to answer your first question. It's going to be more on Mars than on Earth. The second question is yes. So I don't know actually what I keep in mind that every time I've suggested something for one of the rovers, they agreed to send it. But the only meeting that's important to attend at NASA is the last meeting, because that's when they take all the things that they agreed to put on the rover off the rover. And if you think you have been to the last meeting, they will schedule another meeting afterwards. And unless you're resident at JPL, you don't have the ability to fight for what was a laser Raman spectrometer in 1999. We thought we had that on the rover. But they've done a very good job, even though they have been deliberately not looking for life, right? One of which is they have managed to find a hell of a lot of organic materials on the surface of Mars. They are, I mean, this idea that came out of Viking was that the Martian surface was self-sterilizing. You could not have any organics at all, which was obviously false at the time, because Norm Horowitz was fixing organics in the surface of Mars by photosynthesis. And if the Mars were sterilizing, you would have burned those off before you got a chance to look at them. But so the rovers have found a lot of these things. Now, the problem is that they're not set up to look for biomolecules. They're not set up to look for polyelectrolytes. One of the problems is that if you look for molecules that are the products of Darwinian evolution, you'll discover these are also found in asteroids or, you know, non-biological environments. So you can go looking for amino acids, for example. But of course, meteorites have amino acids. So looking for molecules that are required to enable Darwinian evolution is an approach much less prone to false positives than what they're actually set up for. So we don't have anything, and of course the other thing they're not doing is they're not sampling volumes. I mean, what would be nice if Elon Musk were to land on Mars and to pump water to make propellant using your Sabatier process to come back to Earth, we would have a huge astrobiological sample, which Jan could put his device across the stream, concentrate the polyelectrolytes, make sure that they're built from size, shape, regular building blocks, and he'd be in business. But, yeah. Oh,
Yeah, I'm over here.
Okay.
Go ahead.
Hey,
Yeah, hi, yeah, shoot.
Hey, Steve. So does the impress experiment or any assets we have on Mars now able to detect fungal forms like the ones starting life again in reactor room 4 at Chernobyl? The radiosynthesizing fungi, which would love the surface of Mars, I'm sure, because of the radiation that's there.
That's a thought that I have never had. That sounds very interesting. So yeah, put that into the hopper and design something for Jan's Impress mission to get you an easy experiment. You drop 10 penetrators sort of randomly in an area and look for this stuff.
Okay, great.
Okay,
Yeah.
I would like to say the young gentleman in the front, I think your name is Alex, right? You had a very good point about trying to learn from our biosphere. We have many examples of environment which is quite similar to the one which is on Mars, which is anoxygenic, where you have access to carbon dioxide. And none of the life we know on Earth, which is in anoxygenic environment, produces and stores oxygen, and Stevens proposes for Martians. Of course, Steven says that, well, we don't know what Martians do, but since life on Earth haven't discovered this pathway, I think it's quite implausible that Martians will be doing that. So I think you had a very good point there.
Yeah, I mean, your point is, of course, that on the surface of the Earth, which is where you are using the oxygen, you don't need to store it, and deep in the Earth where you don't have access to oxygen, you don't use it at any point in your metabolic system. So Mars has a surface environment where it does have the opportunity to use light for photosynthesis, and therefore would be in the environment which is not present on Earth, where storing oxygen would be useful.
Thank you very much. Just a quick housekeeping note. We will have a couple changes to the plenary schedule this morning. Dr. Vandy Verma had to unfortunately cancel on us. So after the break, we're going to do the Flashline Mars Arctic Station panel, which was originally scheduled as a track talk. So we're going to move that into a plenary slot at 10:45.
you.
Our next speaker now is Dr. Tiffany Vora. She's the VP of Innovation Partnerships at Explore Mars. She's also a Singularity Fellow in Biotechnology at the Singularity University. And she's getting mic'd up right now. Ready to go. Okay. Thank you very much, Tiffany. Welcome to the stage.
Thank you. Good morning, everyone. Delighted to see you again. Thank you to those of you who came out last night for our panel, which went almost twice as long as I was expecting. So I think that's the sign of a good conversation. But today, for something entirely different, I'm going to be pulling further on the biology thread right now and talk to you about harnessing synthetic biology on Mars. And we're very fortunate this year's Mars Society that we have quite a few folks here who are doing this stuff with their own hands. And the next speaker after me, Dr. Erika, she's going to be telling you even more about the work that she's doing. So my job here, Erika, is to set you up. Okay, so imagine I told you about this magical manufacturing technology that is self-perpetuating and self-repairing. It's highly modular. It's flexible in terms of the input that it takes. It is programmable and reprogrammable. It's resilient and it's remarkably stable, that if you keep it cold and dry, it'll keep running and work and wait around for you for years to decades to centuries to millennia to perhaps even more than that. Now, the good news, of course, is we already have that technology. That technology is life. And here on Earth, we've had life for the last 4 billion years, having these properties as this type of system. And so at Explore Mars, we are very interested in the various technology spaces and innovation spaces that are not only pertinent to life off Earth, but also to life on Earth today. Now, I am a molecular biologist by training. I am very interested in the biotechnologies and everything that they touch, which of course in my mind means space. I'm highly biased. But so what we decided to do a couple of years ago at Explore Mars was to spearhead a synthetic biology working group to take a deeper look at what we would actually need in order to further biotechnology and synthetic biology specifically for space, but also with a mandate for making life better on Earth at the same time. These are the members of our working group, and I'm going to be talking today about the findings of our working group. I mentioned Dr. Erika DeBenedictis. She's your next speaker, so I will be talking very lightly about the stuff that she does. We had Dr. Lynn Rothschild from NASA Ames and Brown University, who is one of the pioneers of this field, and Dr. Niels Avrish, who is currently at the University of Florida, who's been doing really, really interesting work with synthetic biology for space, particularly around biopolymers and plastics. So here's why we're so interested in this space today. It's because we're at this almost magical moment in history where, thanks to technological innovation, the boundaries between the living and the non-living world are breaking down. One of the things I'm going to encourage you to do, starting today if you haven't already, is to not think of biology as separate from technology, but rather to think of biology as a technology. And as our exponential technologies continue to become cheaper, faster, more powerful, more accurate, so will our biotechnologies. They're working on the same types of curves. Now, in terms of synthetic biology, biomanufacturing, and biotechnology, we're seeing a push from governments around the world to build up and beef up their bioeconomies. There's lots of reasons for this in terms of things like sustainability, resilience, supply chain resilience in particular, circular ecosystems, all of these things. We've seen really strong plans come out of countries like the US and also, of course, the EU, but lots of other nations around the world, including space-faring nations, have also been working on their bioeconomy plans for in the coming years. We're talking, of course, about countries and organizations like the UK, the UAE, the Saudi government, China, Germany, all have very strong bioeconomy plans. And this is more than just thinking about recycling or more than just biofuel. This is about, again, using biology as a technology in order to make all the stuff that we need for our lives. So there are lots of strong benefits to what I think of as a circular bioeconomy, and I think of it in 3 big buckets. Like, yes, we're talking about protecting the environment, but we're also talking about building our bioeconomies in order to support socioeconomic development and to support industry, right? We're talking about real prosperity here. We're talking about jobs. We're talking about new products and services. And many of these feed very nicely into the UN Sustainable Development Goals, in case that's something that you find exciting. Now, what I always tell people is this is the vision that we have for life here on Earth, but also, of course, this is the vision that we could have for anywhere that humans call home, whether it's low Earth orbit, the moon, Mars, or even further beyond. So, when you think about biotech in space, you're probably thinking about things like these bioregenerative life support systems, which we've seen in play for many, many years now. These are where you've got these closed series of nested closed loops that enable you to use biotechnology and biology to do things like mitigate waste, produce food, keep your air and your water clean, grow your food, that kind of thing. And so I want us to focus today, though, on this component, the microbes. And Stephen set us up really nicely for this. You guys are going to have bacteria on the brain by the time you get to the break. And I, of course, couldn't be more excited about that. So let's focus in a bit more on this idea of what bacteria can do for us in space. Now, they're here as part of this kind of integrated look at microbes in human health and how microbes might be impacting human health in space. Now, of course, for many, many years, the basic idea was microbes in space, bad, right? They make biofilms, they foul up our tubes, they make us sick, they do all these things. But fortunately, we've come to a much more nuanced understanding of how important bacteria are not just to human health, but also to all the stuff that we want to do in these extreme environments. And so I'm going to focus on this bubble right here, these bioengineered microbes, and how we can leverage them in conjunction with our biotechnologies in order to make the things we need. Now, our Explore Mars SynBio working group, we went through the 2020 NASA Technology Taxonomy, and of course, being all biologists, we're like, oh, bio's good for everything. We're like, no, like let's focus a little bit more carefully. And we ended up zooming in on these 3 technology areas that we thought were probably places where the affordances of biotechnology were going to be really strong. Why you should look at using biotic rather than abiotic approaches to solving these problems. And I'm showing you the 3 technology areas here. These are exploration and destination systems, which of course includes in situ resource utilization. We'll come back to that idea. Human health, life support, and habitation systems. And then on the right, material structures, mechanical systems, and manufacturing. And the reason we focused in on these 3 taxonomy areas was because we came up with these sort of 2 big ideas, these 2 big themes about why you would want to use biology for these things. And the first one of these affordances of using these biological systems is it takes you from a take paradigm to a make paradigm. We believe that biology is going to be very powerful for doing things like solving up mass constraints, because again, biology will make more of itself. All you have to do is give it the pieces that it wants and a bit of energy, something to eat, and it'll also make the things that you want it to make. More or less, the biology is complicated. That's why we're biologists, because it's exciting. But that enables you to go, again, from take to make, which will be really, really helpful when we're talking about these excruciatingly long, excruciatingly expensive supply chains. And that, of course, allows you to go from a stored mentality to a fresh mentality, whether you're talking about food, whether you're talking about materials, whether you're talking about medicines, therapeutics, that kind of thing. Being able to get it a small amount of exactly what you need, exactly when you need it, is something that we think biology is particularly well suited to. So now, yesterday, Melody Yashar gave another one of her great talks talking about building on Mars, and she talked a lot about using regolith as a building material. Love it, of course. But Lynn from our working group has also been doing things like working on mycelium biocomposites as a construction material that you could use for Martian habitats. Mycelium being the furry stuff that yeasts grow when you make them really, really happy, or mushrooms do. And actually yesterday on my way to this conference, I stopped off at the BMW Design Laboratory and got to handle a bunch of their mycelium-based leather, right? No cows, just fungus. And it was pretty cool, actually, some really cool stuff. So we're seeing lots of activity in this area about using bio-based materials for a variety of applications. And we would love to see us thinking more about that for space and for Mars. But what I really want to talk about here is synthetic biology, right? So synthetic biology is when we bring together things like genetics, computer science, physics, a bunch of other types of scientific disciplines in order to program or reprogram living things to either make new things or have new functions that they didn't have before, or to improve the functions that biology has already given us. And so the way I want you to think about this, just briefly, is to think about DNA like a source code. In Stephen's last talk, he was talking about RNA, which you could kind of think of as compiled code. But if you want to use that computer analogy, you can also, since I mentioned cars, think of DNA like the chassis of a car that you can add things onto or take things away from in order to change the function. So what we can do with DNA code is we can add modules into the genome of an organism in order to make something new, to improve a function or a molecule that's already there, or even to do something entirely new, like light up if you want your bacterium to act as a sensor, or to move into a particular place. All of that becomes possible using this synthetic biology paradigm. So again, biology as a technology, as a programmable technology. So where do all of these DNA modules come from? The good news is everywhere. Biology has been innovating on Earth for 4 billion years, and that means if we look at the whole kingdom of life, which is up here, we're able now, thanks to super cheap, super fast DNA sequencing, to grab these modules that evolution has already worked on all through the kingdoms of life, right? Whether we're talking about the bacteria that live in your gut, the bacteria that we've been using for 10,000 years to make yogurt, synthetic bacteria that do all of this. And, by the way, this kingdom of life that I'm showing you here, all of this stuff, all at the top, that's all single-celled life, okay? All of the multicellular life, all of the mushrooms and Galapagos tortoises and like me and my kid, we're all the way down here in this very, very tiny corner of the kingdom of life. So we have a ton of inspiration to look to from the natural world in various environments and various forms of life in order to grab these DNA components to try to remanipulate them. But wait, there's more. You might remember that last year's Nobel Prize in Chemistry went to 3 gentlemen who have all been using AI in biology. I love ChatGPT as much as the next girl, don't get me wrong, but I am telling you that the killer app for AI is biology. And the folks from Google DeepMind who won last year's Nobel Prize had worked on a series of algorithms called the AlphaFold algorithms, which first were able to solve to predict a protein's structure from its function. Back when I was a graduate student, which wasn't that long ago, thank you very much, could take you 8 years in order to do that. These folks released a database with structural predictions for every protein known to man, 200 million of them several years ago. It's a big deal. They've also had other algorithms that will do things like predict 3-dimensional molecule structures and how they fit together. They also have one called AlphaProteo, which makes predictions for protein structures that, as far as we know, have never actually been tried on Earth before. And a new version from DeepMind is this AlphaGenome, which is really quite remarkable. It's an algorithm that unifies a whole bunch of types of biological data, again, to make predictions about if I change this in the DNA, this is what I would expect to happen. It's incredible. And remember, Google DeepMind is not the only company working in this space. There are many, 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 many innovators out there looking to harness this AI revolution for biology. A simple example here is an algorithm that came out of a collaboration between the NVIDIA and Stanford, which this is essentially an LLM for writing entire DNA genomes. Now, to my knowledge, none of these completely artificial genomes have been booted up into a living thing. If you know differently, please come find me during the break. But it's a matter of time as we start using these brand new tools to see which ones are actually useful, which ones can we use in order to expand the repertoire of things that become possible when we look beyond what, to our knowledge, has happened thanks to biological evolution here on Earth. So hopefully what I've convinced you in the last 15 minutes is that synthetic biology is awesome, right? So your question should be, what's the holdup, Tiffany? Like, why are we not doing this right now for our Mars missions? So let's take a look a bit more about what's actually needed. So any mission is going to have a particular profile of stuff that it takes with us. Right? Any mission class, whether it's chemicals, whether it's packing material, any of those things are going to all be available as the inputs to a SynBio system, as well as what other in situ resources we have available to us based on where our mission is actually going. And so you've got to take those inputs into consideration when you're figuring out what synthetic biology tools could we be designing and using actually while we're there on the ground, or if we send these missions ahead to prepare for humans coming. Niels has done a lot of really excellent work on this. I'd like to point you toward his papers. I'll be giving you the references down here in the corner, or you can always email me and I'll send you the papers. But there's lots of, it's an interesting chicken and egg problem, right? In order to know what kind of tools we can build for the mission, we need to know what the mission looks like. But we still don't know what the missions look like, right? Like, how much methane are we going to have? How much, we don't know. We don't know these things yet. So we get caught in these loops, but there's some really interesting work that's been done on thinking about how to solve that problem. We also really, really need to mature our current biotechnologies in order to make them useful for these near closed or preferably fully closed loops. Sorry about that. And so here again, I want to zoom in for just a minute on the food and medicine production part of this. We think biology is useful for all of these. But when we again think about food and medicine, I feel like that's where biology really shines here. So Lynn has been working on this idea of an astropharmacy. We don't know what's going to happen to our crews over their 2+ year mission days. So what if instead of having to take every single drug that you could possibly need, oh, and by the way, they may or may not be shelf stable on the way out there, what if you could make, again, exactly what you needed using a very, very small amount of up mass that you bring with you in order to meet all of those potential health, or as many of those potential health situations as possible. And so imagine instead of taking a huge library of these things, you're able to take some bacteria, a bacterial library with you that has been programmed in order to make biological and peptide-based therapeutics, and you wake the bacillus up when you need them, and they go ahead and they make in a small batch on demand exactly the therapeutic that you need, right? So as you say, as Lynn says, you could have a library of thousands and thousands of kinds of drugs engineered into different cells. And we're talking about, not even grams of mass potentially that you would need to take with you for the bacteria themselves. So our working group identified 3 places where we really think technological process and research and development needs to be accelerated in order to bring us to this point. The first has to do with getting shelf-stable reagents. We would really prefer to see things not need to be refrigerated, not need to be frozen, not have to worry about the cold chain. That benefits Earth as well as space, so that would be something we would love to see. We also need to see flight-tested hardware, the bioreactors for actually building, growing, maintaining these bacteria, and then processing what they make in order to purify out what you need. The flight-tested bit is a solvable problem, but it hasn't been solved to our satisfaction yet. And then the last piece that we identified as being ripe for innovation is looking at the chassis organisms, the biological entities themselves that would actually be running this. And there's, as the young man in the front pointed out, there are things we can build on here from evolution on Earth, and that we're really keen on that. There's a whole bunch of considerations about how you choose which microbe you want as your chassis. I'm actually not going to talk about that today. I am going to set Erika up to talk about the amazing work that her group, Pioneer Labs, has been doing for this exact problem. So you get to hear it straight from the horse's mouth instead of from me. So stay tuned for that, please. But what I want to point out here is there's a suite of these technologies here where additional research and development, and specifically dollars, as we talked about a few minutes ago, are needed in order to mature the technologies to advance their TRLs to get to a point where we could actually use them, right? And on the left, you can see we've got a bunch of applications here from plastic and consumable recycling, bioremediation, waste recycling, e-waste recycling, regolith remediation, and then as we talked about those structural components that you could be making with your biomaterials, pharmaceuticals, mining. I mean, there's all kinds of applications here. Some of them are closer to prime time than others, as you can see, and so there's always more work to be done. And I would argue that underlying all of this, what we really need is a strong and powerful and predictive digital infrastructure that enables and is going to empower us to not just do everything ahead of time and bring it with us, but to empower our crews while they're there on Mars in order to solve the problems that come up in front of them when they need them to be solved. And again, biology is a spectacularly nimble system, and it would be a shame to put that affordance down. Also, I really kind of like the idea of the crews having agency on this from a human factors perspective. If you've ever lived in an extreme environment, I was MDRS 3 and 11 and FMARS 10, so I remember I was in Antarctica not too long ago. Actually getting to do the thing that makes your life better is huge for you and for your crew as a team. And so I would love to see us leveraging biology to give us that human factors boost as well. Okay, so if you go and you look at our paper, which we published last year in New Space, you can get a bit of a deeper dive to see how we see that pursuing bioeconomy and synthetic biology on Earth radically empowers what we can manage to make happen in space, on Mars in particular, and vice versa. So last night you heard me talk about how important it is for us to emphasize that innovating for space makes life on Earth better. And I think that synthetic biology is a really fantastic paradigm of that. So please feel free to go deeper there. So the overall vision that I'm selling you here is a world in which biological systems, our 3D printers, our computers, our sensors, our power plants that help us make all the food, fuel, and fiber that we need in order to live resiliently, sustainably, and to thrive and not just survive in the places where we are, right? And that's anywhere that we call home. I'm going to pause here because I reckon there's probably some things you guys want to talk about before we move on. So this is me. This is how we can stay in touch. Feel free to reach out to me on LinkedIn. If you do that, please let me know that we met here. There are a lot of random people on LinkedIn, and I don't want to talk to them. I want to talk to you. This is just kind of the beginning of what's possible here. And I love this. I mean, I'm a biologist. I'm highly biased. But today is the most boring biotech and synthetic biology are ever going to be. It's only going to get better from here. Thank you very much. Let's talk.
To what extent do you think synthetic biology can be used or has already been researched in using it to make organisms more radiation resistant for space travel or life on the surface of Moon or Mars, or to repair radiation damage to the DNA of those organisms?
So we know that folks have been looking at this for quite some time. Biology has in many ways solved radiation problems for us. So if you're looking at bacteria like Deinococcus radiodurans, right, these bacteria that manage to grow inside nuclear reactors, which blows my mind every time I think about it. We have some of those components already from extant biological systems. So the question is, how do we adapt them, move them, modify them, update them so that they are doing the things that we want them to do in these new systems? I reckon you'll hear a bit more about that from Erika. But also with DNA damage and DNA sensing, bacteria are spectacular at this. I actually worked a bit on this in my graduate work a million years ago. So again, what if we could take the stuff that biology, the way biology has already solved those problems and use those? And I have seen lots of folks looking to do that in interesting ways. We can talk more during the break if you'd like, but it's out there. Good morning.
Good morning. Can we create synthetic plasma for blood for humans?
Oh, I mean, yes is the short answer.
But do we do it?
So far, the closest thing I've seen to date has been some really interesting, okay, there's two categories here. I only talked about bacteria today, but there's a great deal of cellular engineering work that's being done to do things like either have off-the-shelf or personalized blood supplies that can be expanded on demand. So that's happening, and also the FDA cleared not too long ago this really cool technique, again, using biology as the technology to grow blood vessels in the laboratory. These are real blood vessels. They're not fake. They're built by human cells. And those folks are using them for the original use case that they used was they deployed them on the battlefield in Ukraine for these horrendous mine-based injuries, that you would rapidly have a supply of these implantable real blood vessels that you could put in people. So looking beyond bacteria, if you're not just talking about using microbial life, if you're talking about human cells, plant cells, animal cells, food, we didn't even talk about food cells, sky's the limit, and it's hard, it's expensive, it's complicated, but really smart people are working on it. Thank you. Yes.
So you mentioned radioendurance in the reactors. What about the fungal blankets covering the reactors outside of the reactors at Chernobyl? NASA extracted some of those materials, sent them up on the ISS, and found that they're radioprotective, but they're also radiosynthesizing. Have you incorporated those observations yet into your studies?
We're not working on that specifically, but I know that there are folks, again, who are looking to use the biology in as many ways as possible. I'd be happy to talk with you more about that during the break, if you'd like. But the idea is to not just be inspired by biology, but to see what we can do to make it work for our purposes as well. Thank you. Yes, in the back.
Thank you, Dr. Vora. That was really outstanding. Thinking big picture, I wonder if synthetic biology could be applied to two significant problems. Firstly, the toxicity of the Martian regolith. Can we kind of engineer microorganisms to fix that for us? And secondly, even terraforming, if we think of the great oxidation event that made our planet the way it was for us, you had cyanobacteria on one of your slides. Could we kind of help the cyanobacteria maybe fix Mars for being better for us?
Erika, do you need me to leave those questions for you? I'm gonna table that because I think Erika's gonna be all over that in the next talk. But yes and yes. Yes, in the back.
Yeah, so I've heard some interesting ideas about, as we go out into different places in the solar system that are obviously very hazardous to humans and other forms of life, that you've got two different ways that you can approach the problem. You can either bring with us adaptations that can conform to what we need to survive, or you can change us somewhat to be able to survive in those environments, right? And obviously this is like very sci-fi to think about, like, how could you edit human biology to survive naturally on the surface of Mars, right? But like, I have heard people say that, there's probably an optimal point of like, how much percentage do you want to do of one versus the other, right? So maybe it's 98% we like take care of, making sure that we have an environment that we need. Maybe 2% we can adapt to maybe some of the most expensive changes. I'm just curious if you've thought about that with synthetic biology, and yeah, if you have anything you can share.
Yeah, so synthetic biology for humans is definitely an area of discussion, shall we say, especially if you give synthetic biologists a couple glasses of wine and then ask them to talk about it. Obviously the issue with human modification is that you have ethical things that you need to talk about that we don't tend to talk about with bacteria, right? We just don't have feelings about bacteria the way we have feelings about human DNA. So there are folks like George Church and Kris Mason who are really out there saying, it would be absurd not to modify humans to be better astronauts, right? Or better Mars inhabitants or deep space or whatever it is. You might even argue that we have a moral imperative to make those folks suffer as little as possible when they're often in an environment that they didn't evolve for, right? Don't forget, Earth is amazing, right? Earth is amazing. We got so lucky with this planet, and our biology has optimized us for this. So I would argue that we should be using every tool at our disposal in order to make it not just about surviving in off-Earth environments, but really thriving there. Again, nobody wants to live on Mars and be miserable, right? No one wants to be sick. So I think these are conversations that we are going to have to be having for many, many years to come before we get to the point where we're willing to have a consensus or to have people say, go ahead and edit my embryos. You guys remember Gattaca? You remember that movie? I still think that's one of the best movies out there for thinking about this. And that's an old movie at this point. But again, happy to have more of these conversations, and they're super important and fun things, fun/terrifying things to think about. One more? Hi.
Hi. For any current students or recent graduates who might want to possibly break into biotech, what sorts of background would you say would be helpful? And what would be your suggestions for them?
My suggestion would be to have a mixture of both wet lab, meaning actual doing the stuff in the laboratory, as well as computational background. Now, when I was a grad student, I had to write all of the algorithms before I could use them for my grad, which was like totally not what I signed up for, but it happened to be that that's the place where I was in my career. I happened to be at the right place at the right time. Now we have this amazing plethora of tools, plus co-pilots like AI co-pilots that can help you write these tools. So I would really encourage every fledgling biologist out there to not consider computation optional. It's your superpower. And the more you can lean into that, I think the more successful you'll be in this world that's coming. Come talk to me more about that during the break. I've got some more ideas for you. Okay, thank you all very much. Thank you. This one, okay.
Yeah, and this one is going to sound like.
I don't have a good.
If you want to just stand up and you can put it on the.
I can also just hold it.
We can do that.
Oh, I can just. Oh, let's do this. I'd rather not. Lots of things on my hand.
Okay, so it'll be, I just want to wrap the cord around. I was worried about what's the cord or the antenna. How does the antenna feel right here?
Just fine.
Okay.
Yeah. It'll just look kind of funny. Totally fine.
Okay,
How's this?
that's perfect for me. How's it feel for you?
That's okay.
We'll go ahead and just get this in here. I can see it. Perfect.
Maybe we can. All right.
Okay, our next speaker is Dr. Erika DeBenedictis, the CEO of Pioneer Labs. Take it away, Erika.
Hello. Well, thank you so much, Tiffany, and also the question in the back for setting me up so amazingly well. I'm Erika DeBenedictis. I'm CEO of a small research org called Pioneer Labs that's engineering microbes for Mars. My background, I started off all the way in computational physics and ended up going as far as it is possible to go in science to experimental biology. And so now I sort of put those two halves back together and think about bioengineering for space science. So I'm CEO of Pioneer Labs. It's a sort of 10-person research nonprofit that is on a mission to make the life we need so that we can build green human civilization on Mars. We opened about a year and a half ago. We're about 10 people. And what's really fun about Pioneer, we're not quite a startup. We're not quite academic. Instead, we get to bring together folks with expertise from sort of both sides of the aisle. So, some of the team is pretty hardcore, like synthetic biology academic researchers. Some of the team is from biotech industry, like Zymergen or Ginkgo, places that have a ton of expertise in engineering microbes like at scale industrially. And so we get to bring those two halves of the coin together. If you haven't read this book, look it up. It's called Braiding Sweetgrass. It's a book by Robin Wall Kimmerer. She's a Native American and a professor of ecology. And I encountered this book maybe about a year ago, and I found it really striking. She talks about how, in her classes of undergraduates, when she asks people to think about the relationship between humanity and nature, most people can't really even imagine a mutually beneficial relationship. And that rings true to me and is striking to her because in sort of Native American philosophy, that's the whole point. That's what all of their origin stories are about, is how humanity is sort of mutually, is like synergistic with the world that they live in. And when I think about space travel, I think it's a huge challenge and also invitation for us to reimagine how we want civilization to work. And I love the idea of challenging us to actually achieve something mutually beneficial when we go to Mars. Girls just want gardens on Mars. I want like less chrome geodesic domes and more us going to this place and learning how to make it thrive, and not just for the humans. And terraforming is one ultimate expression of that possible vision. Terraforming is something people have a ton of, strong reactions to, both positive and negative. But I think within that vision for sort of green Mars, a green human civilization beyond Earth, there are other waypoints that many people find attractive, even before you get to global terraforming. Hopefully, in the near future, right? We're going to have human missions to Mars. We're going to have people on the red planet, probably in pretty isolated, local outposts that largely don't impact the environment around them. In a slightly more distant future, perhaps there's a lot more humans and there's need for just more space, perhaps larger scale agriculture. And you get into this sort of local terraforming, para-terraforming scenario where you've started to build structures that create a favorable environment in increasingly large areas to support larger amounts of human life. And if you sort of take that to its logical conclusion, you end up tiling Mars with greenhouses, and you get to something where you have a thriving planetary scale biosphere, whether that is facilitated by greenhouses or just by positive climate impact externally that allow plants to grow outside. I think all of these, again, people have lots of different visions for what humans' interaction with Mars might look like and what it should look like. But many of these visions involve the same underlying technologies. So it's worth talking about them kind of in the same breath. So what I'm going to do today, I have a talk that's kind of two parts. The first part, I'm going to talk about I'm going to tell you a story that is about one possibility for how you might globally terraform Mars that touches on a bunch of the sort of contemporary technologies that have been proposed for how you might do this over the past sort of 5 or 10 years. And then after that, I'm going to circle back and talk more about what Pioneer Labs is doing specifically to take a useful concrete step toward green technology for Mars that also de-risks some of these longer term sort of terraforming ideas. So what would a globally terraformed Mars look like? Here's one possibility for what might be possible. Humans would enjoy a planetary scale green biosphere. They'd be able to walk around outside without a pressure suit and breathe, thanks to quite a thin atmosphere that's almost entirely composed of oxygen, not of an inert gas. That thin atmosphere would still be sufficient to block pretty much all of the radiation, so there's still no magnetic field. This potential future Mars, the atmosphere is thin, so you have larger day-night temperature swings, and it also is a little colder than Earth. So this particular version of a green Mars, sort of like an alpine climate, you still have permafrost in the ground, but you still can have global plant growth. Now, what's cool about this particular possible future Mars is that it's actually composed of exactly the same raw materials. They're just rearranged. So that potential future Mars doesn't involve crashing asteroids into it. What is different is that two major things have reorganized where the raw materials are. So on the left, we have Mars today. It has a ton of near-surface water ice. This is not even counting possible subsurface water. It has a CO2 ice cap. It has a thin atmosphere, mostly carbon dioxide, and it has materials in the dirt. And on the right is the reorganization of those raw materials that might facilitate the endpoint, possible endpoint I just showed you. So two major changes would transform the planet. Number one, you'd melt the ice, turn it into oceans. And number two, you would use photosynthesis to take a really big chunk of this water and convert it into that oxygen atmosphere with photosynthesis. Photosynthesis takes water, it splits it, It tosses the oxygen away and it stores that hydrogen as sugars, as biomass. Now, generating an atmosphere sort of has been done before, one could argue. The great oxygenation event on Earth happened when photosynthesis evolved. It caused our planet's atmospheric oxygen content to jump 15%. So like generating an oxygen atmosphere at planetary scale is something that we've seen happen before, but it requires a planetary scale biosphere to actually do the photosynthesis. And achieving that requires large scale biomass. And specifically, it requires enough liquid water globally distributed to facilitate a planetary-wide biosphere. So here are two possible options for how you could warm the planet enough to make the biosphere, to make the oxygen, to make the humans walk around outside. One is, greenhouses, solid-state greenhouses. If you have materials on the surface that are both transparent and also insulating, heat will accumulate underneath, right? And that's also a great way to buffer temperature swings. So you can use greenhouses to create environments where liquid water is possible. Edwin, I think, talked yesterday about another option. This is the greenhouse effect, so the gaseous state greenhouse, where you have particles in the atmosphere that reflect heat radiation, thus, again, retaining heat, warming things up, enabling liquid water. And a combination of these two could raise the average global temperature and also buffer temperature swings enough to facilitate a planet-wide biosphere. But before we get too excited about that, you need to grow anything. And Mars is like, famously has basically bleach in the dirt, right? So before you can get to a planet-wide biosphere, even if it's warmer, you need to use ecological succession to build a complex ecosystem, right? Now, on Earth, this happens all the time. So on Earth, when there's a volcano or a forest fire, you end up with these environments that have been basically sterilized by some sort of cataclysm. And the process of ecological succession is the one where a series of organisms in order are able to repopulate that environment, usually beginning with a pioneer species, which is why we're called pioneer labs. Pioneer species are the ones that get there first. They're able to thrive in really harsh environments and do things like convert rock into soil that has enough organic matter that higher order plants can start growing, and you can get a complex ecosystem. Now Mars is so bad that we should probably expect that ecological succession will require human intervention before you get anywhere near things growing on their own outside, likely in the form of beginning with, stirred, heated, protected environments that are, challenge organisms to grow using Martian nutrients and adapting them to tolerate the toxins, but in a quite protected environment that doesn't have to deal with a sort of onslaught of all of these extremes, followed by increasingly minimal greenhouses that do just a little bit of buffering temperature swings or radiation before they can start growing in more natural habitats like ice-covered lakes. Now, this story I just told you is a story. What's cool about the story is we think it might be possible. It's certainly compatible with what we do know about Mars. We know that some of the older versions of sort of how one might terraform the planet, like just nuke the ice cap, like in fact will not work. Please don't nuke Mars. But there's still a lot of outstanding research questions. So if you follow this link, this document actually, this slide deck corresponds to a bigger document that breaks out all of those things I just described in more detail. There's an appendix where you can sort of like look at some of the math and like follow the references. And it also talks about what the outstanding technical questions are, like the real basic science questions that might mean this isn't possible. It talks about other methods that have been proposed for terraforming and also the more immediate benefits of studying really any of this. And this document for reference was created as like a handout to get folks up to speed at a workshop, the Green Mars workshop that we hosted last month. It's the second time we've hosted a workshop on this topic. And what's cool is like the workshop doubled in size since last year. And also the number of people who like actually full-time have the ability to work on these sort of Green Mars technologies has like 5x, largely thanks to Edwin and his team, and like my team got bigger. And we're sort of trying to figure out how to just more broadly get there to be more research funding, because there's more problems than there are people to work on it right now. But in general, it's pretty cool. It's like terraforming used to be solidly a sci-fi concept, and now it is becoming a research discipline where we can actually pick it apart and interrogate, is it feasible? How would you do it? How long would it take? How much would it cost? Which is really cool. So for the rest of the talk, I want to circle back around and talk more about Pioneer Labs and what we're working on now. So our goal is to create the organisms, the pioneer species that would be necessary to make Mars a nicer place for more life to live. That starts again with the very first microbe that you might actually deliberately use on Mars to do something useful. And to be really concrete, we spend most of our time thinking about what could you possibly flight test in like the 2031 launch window that would be independently useful and also de-risk every, a ton of the basic science questions that we have about things that happen to the right. And we spend the minority of our time thinking about how does that first flight test facilitate or tie in to some of these larger terraforming endpoint visions. Now, if you're going to do a flight test, it's going to be very expensive. You have to have a really good reason to do it. So we have tried to spec out what a possible mission in 2031 would look like. And I'll describe it with sort of two sets of requirements. So on the left, no matter what you're growing, what we know about today is carbon-based, water-based life. And so there are some non-negotiable universal atomic requirements for the materials that you need to have on hand to grow that life. Your life needs to get carbon, nitrogen, phosphorus, sulfur, and then like a long tail of like little extra heavy metals and stuff like that in order to grow. And ideally, in order to work on Mars, you want those raw materials to be sourced locally. You want to do ISRU rather than bringing everything with you, because then what's the point? On the right, there has to be a reason for this to happen. So an ideal 2031 flight test would be useful. It would have some sort of useful outcome for either robotic or human missions. It would de-risk the fundamental science. Do we really know what's in the soil? Like, is there an impact of growing in slightly lower gravity, stuff like that? And then also very crucially, this is now not actually that far away. There's going to be ongoing search for life missions. And it's important that at this stage, any organisms we deliberately send to Mars really actually cannot terraform the planet. They can't grow outside. They really are, fully domesticated and need us and our artificial environments to grow. Roughly what we've come up with is the idea that you would send a sort of refrigerator-sized unit to Mars. It would take as inputs totally raw materials, dirt, water, and air. It would use those raw materials as food to grow microbes, and those microbes would produce a useful product, bioplastic, which you can use to make greenhouses. So this is a cool way to sort of bootstrap to that, to the right on that ecological succession image I showed you. So what have we done so far? So last fall, we started with this question: what is in the dirt in units that like biologists use? There are a lot of regolith simulants out there. They are mostly designed to mimic like the material properties of regolith. So like testing your rover tires, it is not obvious whether or not they are suitable to mimic the chemical properties of regolith in the ways that matter for biology. And so what we set out to do was develop a defined media recipe that mimics what happens when you put basically like a tablespoon of regolith in a liter of water and you shake it around and you get the solute out of it. And so we put that recipe together by looking at all the sources of data that exist, direct measurements from Phoenix WetChem, as well as like indirect measurements of terrestrial, simulant leachates and other things like that. And so what we came up with was soluble Mars regolith media recipe, which interestingly actually has a bunch of good stuff in it. So it has fixed nitrogen. That's great. We needed nitrogen from somewhere. It has phosphorus. That's great. That is in fact the only place you can get phosphorus. So if your organisms can't use that, you're out of luck for growing things. It has a lot of the sort of trace elements that you might require. So Mars dirt actually has a lot of nutrients in it, but it also has toxins. So it has perchlorate, which is famously, in general, not great for growing things. It's basically bleach. And it's also very salty, which, you used to like sow your enemy's fields in salt so that they were screwed. So, I mean, it's not good, right? And the challenge with growing stuff in this is like, can you make organisms that can tolerate the toxins enough that they can use enough dirt to get the nutrients out of it? Now notably on that recipe, there wasn't any carbon, which is fortunately in some ways the easy part, right? So Mars has a thin, but basically totally carbon dioxide atmosphere, which is great. There's two kind of obvious ways to use that carbon, give that carbon to life. One of them is photosynthesis. That's what we're used to. Plants breathe carbon dioxide. That's what they make that's their carbon source. The other option is electrochemical carbon fixation. So what that means is that you get an air compressor, you feed CO2 over an electrode, and you use solar panel energy or whatever to turn that gas into vinegar, basically, formator acetate. And then you feed the vinegar to an organism as a food source, rather than forcing the organism to breathe the carbon dioxide itself. And per surface area, electrochemical carbon fixation is way more efficient. Photosynthesis is actually not that efficient per surface area. It's way less good than solar panels. And so that's the route that we're imagining going for this initial, refrigerator-sized box that is size limited. The other benefit there is that obviously, if you're feeding an organism vinegar and it needs that to survive, there's not like vinegar elsewhere on Mars, so it cannot grow outside. It is not an autotroph. And so that's one of many great ways to ensure that this complies with planetary protection. There's no chance these critters could live outside. Oh, and this is there do exist of folks who have built these units already. So Electrolist is one example of a company that has sort of like a microwave-sized box that you put in a CO2 line and out comes acetate, which is our carbon feedstock of choice. So now knowing what we want this organism to eat, the next question is like, which one? So we nominated a whole bunch of different organisms to try out. This is a subsampling of them, and we tested whether they can use our carbon source of choice, acetate. Can they use the nitrogen and phosphorus in the chemical form that they come in the regolith, yes or no? And then also, do they die immediately at these sort of salt concentrations and perchlorate concentrations that they're gonna have to face to get enough nutrients? And we screened a bunch of organisms against this incredibly simple set of requirements. So interestingly, I was expecting that we would screen a bunch of organisms and the extremophiles would do quite well. There's tons of interesting critters that in theory, Deinococcus radiodurans is everyone's poster child, but I mean, there's a ton of organisms that one might think would work well. Actually, what we found is that Pseudomonas putida and Cuprovitus necator, which are they're not quite E. coli in how model organism they are, but they're the usual suspects for doing biomanufacturing, they actually do better. And I think that's because we're still screening them in the context of a stirred heated bioreactor. We're just giving them like kind of a weird feedstock. And those are the organisms that you use in that scenario. So I think we're not going to get to extremophiles being truly better until we think more about real outdoor, like more greenhouse-like conditions. Okay, so we know what to feed them. We know which organisms are promising. What do we make first? So Tiffany gave a great overview of the enormous number of possibilities for how to apply biotech on Mars. And indeed, I think that is in some ways the problem. We're so under-constrained in what to produce that there's some decision paralysis in the field about what to do first. So we tried to really break it down. We considered a ton of possibilities. Here's a subsample. We basically wanted to find something useful and dead simple. Like, what is the first thing to do? The current goal is to do perchlorate soil remediation. That's a one-step process. Remediated soil is useful. Great. The sort of stretch goal is to do this bioplastic, which again is a one-step process, but it does require a little bit of post-processing to actually purify the bioplastic out of the organism. And so that's our sort of stretch goal. And we'll leave things like food for later. Food would truly require a multi-step process where first you remediate perchlorate, and then you use that raw material to produce food for humans. So that comes later along with a ton of other possibilities. So putting it all together. Here's a cool first example of sort of putting this all together. This is growth in 100% Mars atoms. So water, the soluble regolith media recipe, which is water plus Mars dirt plus acetate, which you could make on Mars. This is growth of wild type C. necator. It does not grow well, but it does grow. And then here's a bunch of engineered variants that we've created. There's a whole other talk I could give about strain engineering to improve how well organisms can use Mars juice, the soluble regolith media. And we've already, even with doing just a little strain engineering, we have improved variants that grow to much higher total biomass and much faster, which is great. From some of those engineered strains, we have actually done confirmation that we're making PHB bioplastic. So we purified a mystery white powder out of our biomass, and we actually tested it with NMR. Indeed, it's bioplastic, pretty cool. So this is a cool, sort of very minimal first example of actually improving generation of a useful product from total Mars atoms. So looking back at this specification, this checks the boxes. On the requirement side, we can get everything we need for organisms to grow directly from materials. On Mars, we have to do very, very little processing of those materials to make a feedstock that is usable. In terms of, a reason to be for a flight test, yeah, if we're making bioplastic, that's great. That'd be useful, for robotic or human missions. That's just a building material you need in bulk. This is a great way to de-risk the science. Growing anything on Mars in real water plus Mars dirt would be an enormous step forward for figuring out if we have the recipe right, how does gravity impact things. I mean, there's a huge number of things that need to be de-risked. And yeah, these organisms, they will be dead 10 different ways if they get out of the bioreactor. They're sterilized by freeze thaw, by radiation, by desiccation. They won't have anything to eat. And all of that can be very thoroughly benchmarked, so we can be very certain that we are not going to accidentally contaminate Mars by doing this on purpose. Early days, it's still early days. There's a lot more work to be done. We do need to build out a full techno-economic analysis of the yield of this box. How big should the bioreactor be relative to the carbon fixation unit? Questions like that, like how much does it produce per 24 hours? These are all things that we need to actually get to the next level of detail of modeling before we can, actually start designing stuff. I'd say I glossed over a detail, which is that to purify bioplastic, you do need to do a purification process. We think that can be done with no consumables, no consumable reagents, or very, very low consumables, but we haven't actually optimized that process yet, so that remains to be done. And I'd say also PHB bioplastic, it's like the least good bioplastic. It's the easiest one, it's the right place to start, but there's a much bigger world of possible bioplastic alternatives or flavors of bioplastic PHAs and stuff that might be better for making greenhouses. There's also cellulose acetate and other materials that could be produced with basically a very similar process. And we'd like to more fully explore that space of possible output materials. Again, we're a small research nonprofit, so we do open publishing. We publish everything through Substack. We do these sort of like semi-technical essays. Most of what I talked about today comes from these two Substacks that you can look up. One about how we made the Mars dirt recipe, and one about how we did this sort of scoping for what to produce. Those two were led by Una and Harley, along with the whole team, but shout out to them for doing super awesome job on it. And again, thanks to funders, this work at Pioneer is primarily funded by the Acera Institute's residency program, but Pioneer more broadly also has grants from Schmidt Futures, the Align Foundation, and others. And also thanks to Nils and Edwin and many others, a great community. Thank you, Tiffany, for setting me up. And yeah, happy to take questions if we have time.
Can you go back to your slide where you have your recipe for the soil? I'm trying to get everybody in the room to design their own impress penetrator. And basically, they should look at that particular list and show everything that's wrong with it. So one of your problems is you have perchlorate, that is, but then you have bleach. The literature on bleach comes from Richard Quinn, who had a high radiation intensity source to make the bleach, but he did not consider the processes by which the bleach self-destructs. So if you go to Walmart and buy yourself a bleach container, you look at the expiration date, because bleach reacts with bleach to make not bleach and makes relatively not. So Richard, if you only consider how you make bleach in your Martian model, but don't consider how you destroy bleach, you have a lot of bleach. So second, the chemists will always rebel when you say perchlorate and bleach are the same thing. They are not. They're extremely different. Bleach could have been the non-biological reagent that gave Gil Levin a false positive for life. If there's bleach there, I can explain Gil's result without any life at all. But perchlorate, you can't. And second thing, calcium at 1.17 millimolar and phosphorus at 0.1 millimolar, those two precipitate. So one of your problems in biology is if I have life and I have appetite, calcium phosphate available, one of the major challenges to living systems is how do you get phosphate out of appetite rock, since it's so insoluble. So you must see a precipitate coming.
Yeah, I'm happy to talk more. Yeah, so we had a whole Mars milk disaster for a while where, I mean, the fundamental difficulty here is A, there's like limited data on what is in regolith, and B, the chemistry of what happens when you add water depends on the pH at which you are doing that leaching, as well as the time scale at which you're doing that leaching. And yes, things precipitate. And so those things have to be balanced. So there's a whole like deeper dive on like, in order to get, for example, enough of the metals that you want your microbes to have, should you do some leaching at like an acidic pH, which would also pull out more phosphorus, some leaching at a neutral pH, and then mix those two, for example.
Right. The other thing is you're not looking at redox balance. So that nitrate +5 oxidation state to become ammonia, the interesting stuff, you need 8 electrons per nitrogen atom.
Let's talk later, because agreed, the chemistry of this formulation and its like shelf stability and stuff like that, it's like a whole, it's tough. Yeah.
I was wondering about the statement that everything is there that you need for global terraforming. You certainly have carbon and nitrogen, but I was wondering about the amounts that you have, because the atmosphere is very thin. And you have some nitrogen in the soil, but if you wanted to create, let's say, 1/3 of Earth's sea level pressure atmosphere, and you want to have enough carbon and nitrogen to have eventually plant growth over a large part of the surface, Is there enough carbon and nitrogen in the atmosphere and the top surface as far as we know today, or would that have to be imported eventually?
Carbon, yes, nitrogen, no. And this is why the story I told you doesn't involve nitrogen in the atmosphere. It involves quite trace nitrogen in the atmosphere. And I think that's, I mean, yeah, if you are attached to having an inert gas in the atmosphere, like it needs to come from somewhere, and it doesn't seem like there's an obvious source of inert gas already on Mars. And so you would have to figure out how to import it. At the same time, it's not obvious whether you need an inert gas in the atmosphere. You could just have low pressure oxygen, which is what like fighter pilots breathe, right? So it's unclear. And I think that there's a bunch of outstanding questions about, you know, exactly how much low pressure oxygen can you have that would be enough to support human breathing, but low enough that it's below like the flammability limit. And it's like, you know, it's like these are measurements no one has taken. We have some estimates, but that's like one of the big areas of ongoing research.
I mean, if you had an atmosphere that was, say, 35% ground level pressure and was half oxygen, it would probably be breathable. But I'm wondering if that atmosphere being 50% oxygen, that thin atmosphere, if the flammability would be an issue if you have that as your global terraforming plan.
Yeah, like you should look up, if you look up the, I think that this is a great question, and I think the answer is like we don't necessarily know, but if you look up this one, and there's an appendix that has some citations for like estimates of like where would the flammability limit be for pretty close to pure oxygen and stuff like that. But yeah, I think that's a great question. That's one of the things. It's like I think we don't know quite where the flammability limit is.
Well, the Apollo missions were conducted in pure oxygen, low pressure atmospheres in the cabin. So yes, you might have more proclivity for fires to start, but it's not like everything just goes on fire all the time.
Quite so, but Apollo was also, it was like very designed to have all the materials not be flammable, and I think it's a...
I believe that Skylab had a 2/3 oxygen atmosphere that was 1/3 total.
Mm-hmm. Oh, that's a microphone.
So that's a mostly oxygen, low pressure atmosphere that they lived in for months.
Yep. There's a separate issue, which is like the nitrogen source. There's fixed nitrogen in the soil, so you can just use that to create biomass. If your biomass is then degrading, it may release nitrogen back as a gas, and then you have to refix it. And nitrogen fixation is less efficient at like low pressure. So there's like a separate issue of like there has to be some nitrogen cycling through the atmosphere, but could it occur at like much lower pressure? Unclear.
Question on your regolith recipe, and particularly with production of something like bioplastics. I would imagine that those are just kind of averages, but have you played with what are the critical components in that recipe and the likely large variability you're going to find in different regions on the surface?
Yeah, so great question. This regolith recipe is mimicking what is in the fines. So Mars has like global dust storms, and the component of the dirt that is like quite the fine particulate size is thought to be pretty evenly globally distributed and homogeneous. That's not the same as the rocks. So obviously, if you have rocks that are made by geologic processes, maybe you have like a vein that has more of some metal or something. So we're trying to make a recipe based on what we think is a pretty safe assumption about a material that will be pretty ubiquitous, specifically for this reason, which is that like you have no idea what the rocks will be like where you land. Separately, yeah, so the right. So then on top of that, then we're doing these tests where it's like, okay, if you vary the actual concentration of anything essential up by 10x or down by 10x, are you screwed? Right? And the answer is, so far, it actually looks like that's not necessarily what's limiting in this case, and more what's limiting in this case for these organisms in this particular bioreactor context is that they're trying to tolerate both this like sort of weird feedstock that's like not super nutrient rich and like also has like some like salt and stuff like that, while also tolerating acetate, which is itself a stressor. And so the contribution of individual things in the regolith recipe seems to not be what dominates. It's like not a deal breaker. There's no one thing that's a deal breaker. That may be not true in a different context where you're growing it with photosynthesis, for example, or for different organisms. But totally, that's exactly the way to do it, is like, you know, we can only do so well, we only know so much about the dirt. So when we have existential uncertainty, let's experimentally vary everything and see how concerned we should be.
Is there a source of material that you could use to extract deuterium for fusion?
Deuterium for fusion.
Heavy hydrogen.
Yeah, water.
Yeah.
But is there a place, a location that would be optimal for colonization and mining?
I mean, I think in that case, well, I mean, people are already picking sites based on where they think near subsurface water will be present. So, I mean, maybe that's the answer there. But yeah, I'm not sure. It's a good question.
You said it's the least useful bioplastic. What applications would it have in its current form? Is it good for containers, or can it be recycled? Can it be used as a feedstock or something else?
Yeah, you can like 3D print with it, and you can redegrade it. All the bioplastics are biodegradable, so you can reuse it. It's like least good in the sense that it's, I mean, relative to other types of bioplastic like PHAs, which is a whole nother related class, it's just like, it doesn't necessarily have as good material properties for, you know, flexibility and stuff like that. It's also, for the greenhouse example, PHBs are not transparent. And so you would want to use something like cellulose acetate, which is like what which is like what glasses are made of, or something like that. But the space, there's a great graph, I don't have it in my deck, but there's a wonderful graph of like, there are so many different types of bioplastic materials, and they all fall at different spots in terms of like, you know, flexibility, like how brittle are they, how strong are they, and then they have different applications. But there's like a ton of variation, and it can mostly be synthetic biology programmed by quite what genes you put in your organism.
Thank you very much.
Oh, that's something else.
Okay, do you guys want a little break? We're kind of running a little late here. Do we want a short break, or should we just keep rolling? Short break? Okay, we'll take a 10-minute break then, and we'll come back. The next talk is going to be the Flashline update with myself and Andy Greco.
Not forever. I'll be around for another
All right.
I have you for...
Sure, yeah. No, yeah.
I found a slot for you. 4:30.
I guess there was a decision.
I guess so.
You were inspired. You're gonna build something for your press.
Raw material. They degrade it somewhat.
You are.
No, they would also, yeah.
I just wanted to share some of our recent literature.
Yeah, I'm not sure about that either.
You want to be with us?
Other collaborators done some more work on like different bioplastics, and he like actually there's like some data in the literature about like how fast they degrade under UV, and he like remeasured it and found it was just like completely wrong, or it's like, you know, it's like more nuanced. So
Well, and sunlight on Mars, sunlight on Earth.
not quite the same, and yeah. Totally.
Nice to see you.
Yeah, I mean, I think a lot of these, it's unclear. There's also there's a lot of interest in using like aerogels, either like cellulose-based aerogels or like silicone-based aerogels, and similarly, I don't know how they degrade their degradation properties either. So might need to replace them. So I
had one question after.
Thank you.
What you're
Let me go with this. What's up?
I also did DNA get to have this combination of information is so
cool.
intricate, so.
Yes.
Yeah, we're filling time.
So.
Hi. Yes, hey, nice to.
Not what I thought was going to happen, but it's very.
If you can email them to me.
Hi,
Oh, yeah.
my name is Dennis.
Hi, Dennis.
Fantastic, by the way. A little background on myself. I've built spacecraft for like the past 20 years. I'm retired right now. So right now, very interested in terms of like your mission and your mission planning and kind of where you're going. Like how far along do you think it's going to take to really design a mission?
I think we're trying to grapple with that right now. I mean, I think
Hi again.
the current team is designed to make microbes. And I think beyond that, we can make the bioprocess. I think doing a flight test and finding all the hardware necessary for it, especially given that
No, I don't think so. Should I?
there are very limited options for bioreactors that have like there's no heritage targets. Is a big deal, and so we're looking at options for like our job is to provide organisms and like designs, and then we work with other folks who are like more.
So that's kind of what I'm kind of doing right now is connecting them together.
Question.
So,
Yeah, so we go outside
or keep in touch with you. Because right now I actually have a meeting with APL in 2 weeks to talk about.
Yeah, super cool. Yeah, we've talked to some implementation partners. I've also talked to like Adams and Ambrosia. I mean, there's like a handful of these floating around. I think it's not obvious to me. Like actually on one hand, we're trying to keep it as simple as possible. Like we're not trying to implement
Right.
So it's also difficult. On the other hand, there is still a lot, just like basically all like the things we need to be made, and so it's a real.
So then here's the $1 million question. From funding we're doing it.
Yeah, so we're funded as a focused research organization, so we have like science collective money basically. We're mostly supported by the Sarah Institute, which is a nonprofit that Jed McCaleb started. He also is the for-profit founder of VAST, which is where you do space station. And we have funding for the next, year and a half at least, but specifically to do. I mean, not like it's not funding at the scale that would be necessary to do a flight. And so there's a outstanding question of like who funds that.
Okay, that could be something we might be able to help you out.
Yeah, cool. I'd love to talk more. Yeah.
Absolutely, I'll be in touch.
Yeah.
Okay, thank you.
Hello.
So I'm giving a talk at one about building nuclear reactors on Mars, but as part of that research, I ran across this one paper from researchers in Spain where they were growing algae in a solution with depleted uranium and found it basically enriched it by a really impressive amount. And I've never seen anyone try to replicate that as far as I can tell.
I haven't heard of that paper. I'd love for you to send it to me. My email's on it.
It seems like it's pretty simple. There's a lot of. If you have a mass.
Just go for it.
Well, I mean, where are you going to get the material? But yes.
Well, it's depleted.
As
far as I know,
you could send it to me, that'd be great. I'd say though, like, I mean, Tiffany might have even mentioned this. There was this, people found this like radiotropic fungus at Chernobyl that they thought was like growing, like they observed it growing toward radiations, like putting its little hyphae out in the direction that radiation was coming. And the hypothesis was it was like somehow getting energy out of that. But then people tried to replicate it and they couldn't. And then as it so happens, like actually the original sample that was actually collected at Chernobyl has been lost, and now there's a war in Ukraine. And like when you get a fungus that's like technically the same species, but it's like not actually from Chernobyl, it doesn't do this. So this is all just to say, I'd be super interested to read a paper. As you say, if it's an easy experiment, like we could do it. But it's like. It can be hard to know how to replicate an experiment like this because you like don't know what it was about the depleted stuff that mattered or something like that, or like what the sample was. So, but yeah, super cool. We're following up on it.
someone needs to try this again.
Yep. No, send me the picture. Nice to meet you.
I did.
Hello.
I threw a
Yeah,
dart.
nice to finally meet you. Yeah. Yeah.
So as I was thinking about your presentation, I was wondering if we could use regolith analysis where we might use a rover or something like that. We can test it. The question is, if you're not within the biosphere, you're actually running outside of the biosphere. See what the reactions are. What does that look like? The question is, there a risk if you're actually doing something outside of the biosphere in the term process?
Wait, tell me what do you, tell me more.
If I think
Yeah.
Maybe you can help Virgil and guide me around.
Oh, like outside of a heated environment?
Right.
Yeah.
So what are the
I mean, you should talk to Erika, right?
Like planetary protection risk
Yeah.
or like.
It's not taking protection, but as well.
Readings properly.
I saw, but I was
One and missed.
Yeah.
Everyone's here.
Yeah, no,
We'll talk when you-
well said.
Great.
I mean, I think, okay, the planetary protection wise, I think it depends a lot. Outside on Mars, like Mars, most places on Mars are below the triple point of water. So like the water's unstable, so it'll either freeze or like if you put an organism outside, it would get freeze dried, right? Which is not a good recipe. It's not a recipe for stuff like growing exponentially. That said, I mean like things can be freeze dried and they're still biochips.
And solutions freeze at a slightly lower temperature.
Sometimes, yeah, sometimes. And it's also very salty, and so they'll further freeze at a lower temperature. So you can have brines that are liquid at 6 times a year. I mean, I think in general, there's like a really big difference between like putting an organism on Mars in a setting where it can like temporarily tolerate or survive like the extremes of the outdoors versus putting an organism and expecting it to thrive and grow and make biomass. Those are super different.
Right.
Perfect lab.
Okay. Yeah. True.
Yeah.
Maybe. I was just telling someone else the
Oh, there's
anecdote.
no maybe about it, about the radiosynthesizer. My hypothesis is, but they know that there's radiosynthesizing.
Yeah, it can't get replicated, but sorry. I can't remember the names. They're the radiotrophic fungus in Chernobyl, like it's like unclear if it actually gets energy out of radiation. That's a very different thing, but yes, totally.
It's a very different thing. Lord now having expansion. Down to the electromagnetic spectrum.
Yeah, it would be ideal if there's like it'd be very cool and a very cool like planetary protection thing if you could have organisms that like eat radiation that you only get on Mars because then they could also not grow on Earth.
That's it. That's what we got.
Yeah.
Reattracted humans.
Yeah, not can't get replicated, but well, and in fact, well, or like people obtain species, and it's like they don't even see it being radiotrophic. It's also they obtain the same.
Okay, everybody, we're gonna start up so we can keep on our tight schedule today. Got some great talks coming up.
And the original Chernobyl.
So if you wouldn't mind taking your seats, taking conversations outside, that would be great.
Thank you.
Come on, I'm going to welcome up to the stage Andy Greco, our chief engineer of the Flashline Mars Arctic Research Station. We're going to go ahead and get started, everyone. So if you wouldn't mind taking the conversations outside, that would be great.
So you're going to ask for biologist or a theoretical physicist or what's happening.
I'm a lawyer by training.
A lawyer?
Everyone's having a great time. I can see it. I love it. I love it. We worked so hard to put this event on.
And it has been excellent.
Okay. So we're going to get rolling with this panel. We kind of threw this together last minute because we had a cancellation. So we're going to talk a little bit about Flashline. With me on stage here is Andy Greco, our chief engineer. I'd also like to welcome to the stage Dr. Laura Thomas, who is one of our crew members this field season. And so let me run over here to the slide deck. So this comes in the context of 25 years of being up in Devon Island. The Mars Society built Flashline in the year 2001. There's a book by Robert Zubrin, Mars on Earth, that I tell all the crew members it's required reading because it tells the story of not only how we built the station, and it did not go according to plan, building the station, but also why we built the station and why we do analog research to begin with. Mars on Earth is an excellent book. It's an excellent read. It's an excellent resource. It's over 25 years old now, but it's still highly relevant to what we do. And it also talks about MDRS in the first cruise to MDRS. But Andy has been part of our last 3 field seasons. We had a hiatus for 7 years where we didn't send crews up to Flashline, and that ended 3 years ago when we sent crew 15 with Andy and Terry Trevino and Andrew Wheeler from Australia, Caleb Poole and Olivia Drayson. They reactivated our station. And then last year we sent another crew. And then this year we actually sent 3 crews. We did a field season with 3 different crews. Andy led the advanced team. And then we had 2 science crews. Laura was on the first of the 2. And so we now see this program as sustainable. We see this program as something we plan to do every year. It's an excellent opportunity for not only scientists to study the pristine environment up at Houghton Crater. It's a direct hit impact crater. There's only a few of those on Earth. And it's also a pristine environment to do things like climate change research. I mean, there was a study we did this year on bioplastics. And so it remains a highly relevant scientific resource. And we have lots of interest in this program, and we're also looking for folks to help with mission support. We're looking for folks to help with becoming crew members in the future. So, this is a program that we hope everyone can be part of in some way, even if you're on the sidelines right now. So,
Yeah.
Andy, I'll turn it over to you
Sure.
for some comments.
Yeah. As many of you know, Flashline is one of the higher echelons for remote research stations, being up in the high Arctic and being so far away from resources. There are 2 eras kind of the station that James touched on, the before that break and afterwards. So many years being uncrewed, we actually didn't know what the status of the habitat was. We didn't know if it was structurally sound. We didn't know what the systems were like inside, if anything was usable. So planning that first, that return to the station in 2023 took a lot of effort and a lot of dipping into the archives and reading all as much as we could about the development of Flashline, and of course the book Mars on Earth. The copy that I have is just dog-eared and notes scribbled in it and post-it notes sticking out of it. It's been an excellent resource. And as far as resources goes, the research that we're doing, hopefully we'll get into the JSI, the Journal of Space Analog Research. And the future of this, you can see we're getting more and more frequent. We're getting more and more people, more research going on, but we need help. So this was the first project that the Mars Society put on.
and it went amazing, except for, you know, some of the hiccups. But the overcoming of those challenges of how to build this, and if you don't know what I'm talking about, I encourage you to read Mars on Earth. That is an incredible story. We need people like you. We need volunteers. We need help. We need smart people. It's not just a few people standing in front of you here that can make this happen. So one of the things that I wanted to ask or make a call to is if you are interested in this, if you have something to share, please reach out.
And Laura, can you tell everyone a little bit about the importance of the science at the station and what you hope to, you know, it brings to the community?
Yeah, so we had some great projects running there this year. I'm a psychologist, so my project was around bringing an AI-powered wellness tool to help track well-being in a way that was maybe less intrusive than questionnaires. But we had a great project on nanoplastics, as James mentioned as well, and also extracting water from gypsum present. And one of the things that I'm really passionate about is that that knowledge doesn't just stay and sit within circles like this. It's also important that that's getting transferred and communicated with the local communities, and that we're working really closely alongside them to achieve that. So it was really meaningful that we had, for instance, an outreach session with the local communities there, that we could talk about this and disseminate that knowledge. Yeah.
So some of the challenges, Andy, you want to talk about this? And I love this quote from our friend Anastasia Stepanova.
Yeah.
She was on we had a crew in 2017, which was the last crew before the hiatus. That crew was called Mars 160 because they spent 80 days at MDRS and they're intending to spend 80 days at Flashline. The way it worked out is they spent about a month at Flashline. But she, you know, she's quite a character, and you know, I think that quote sums up a few things.
Sure. And the quote that island does not care about you, it's not necessarily negative. It is an uncaring place. There are no resources. There is nobody there to help you. You bring the challenges and the solutions with you, and when you go there. And these are just some of the images of the things that we had to deal with. Connectors that were not working properly, so we had to manufacture solutions to complete a circuit. ATV repair, water system leaks and repairs, electrical work. And yeah, another photo of the new hot water heater that we had to replace because it ruptured after a 2023 expedition. So 2024 crew didn't have hot water. Sorry, guys.
And I mean, it's worth mentioning that the station now is in the best shape it's ever been. You know, we've gotten a lot of the equipment upgraded. Everything's been tried out, and we now have 3 years of crews using everything.
Yeah, it's incredibly robust. I don't know if anybody in the room was there when it was designed, but you did a great job. It is very stout. It's not going anywhere. It doesn't shake in the wind or anything like that. And it has handled 25 years very well. There's no reason why it can't go another 25 or more with proper care. And some of the things that we need, like I said, we need people who will care for the island and for the station. Expedition planning is a big deal. It starts in January, and there's even talks about that being too late because we go from January right up until the mission for expedition planning.
I think we should start planning this like next week.
I have no problem with that. And to think like that we haven't started already is not correct. We're already thinking about it. And maintenance engineering is a big one. Favoring less destructive activities while we're up there, cleaning what we have, and planning for the upcoming research seasons.
Now we're up in Nunavut, which is governed by the native tribe, and so it's very important for us that we maintain a good relationship with them, and we have. We've been able to get, as part of the program, we have to apply for many different types of permits to operate up there, for both the tribe and also the Canadian government, the Crown government, because we're on Crown land, they call it, but we are in Nunavut, which is governed by the Nunavut tribe. And so outreaching to them has been a priority for us, and you know, and it remains a very important thing that we include them in planning. We offer them opportunities to be part of the science we're doing, to be even provide crew members in the future. We were going to have a crew member this field season from Resolute, but unfortunately that person was not able to make it. But in the future, that is, you know, absolutely an opportunity we want to provide to them and to the local community. Laura, do you have any thoughts on working with the local community?
Yeah. So that cross-cultural awareness that you're talking about there is so critical. It's one of the competencies that NASA names as being integral for human behavior and performance. And I mentioned previously about the outreach session that we had, and that was really special for me because we got a huge turnout of people, and it was a way for me of clearly demonstrating that the Mars Society really cares about that and is working very, very hard to work with and share those knowledge systems. You know, I'm British, as you can hear, but I do live in Canada, and those connections with Indigenous peoples are really, really important. So the Inuit up in Resolute are just incredible, and the connections that we have with them are amazing as well, and I'm sure that they will continue into the future.
So this is our last slide. So we do have a track talk today at 2:30, which we'll be going into a lot more detail on the field season that was just completed. We encourage you guys to attend that. Any last comments, Andy?
Nope, just I really like this photo because of the last 3 years, this was, I think, the only year that we saw accumulating snow on the ground in the Arctic. 160 was trying to be there for 80 days, but that is an enormous challenge. It might not sound like it, but we only get a short window where the sunlight is there for 24 hours a day, never sets, and we have to get in and get out before the winter comes. So that's part of the big challenge we face, but we had some interesting weather this year.
Okay, we have time for maybe one or two quick questions before our next speaker. Go ahead, Eric.
I'll
Just shout it out.
Yeah, I'll just shout it out. So Eric Christoph with the EVA Link project, Society of Chicago. Already talked to Terry about getting our system up there for next year. So backing down, figure out all the constraints and stuff like that. So we're super excited to try to do this for Flashline. Question for you guys. That's come up with the crew. It's probably before. Has there been any kind of interactions or collaborations in the past with NASA's Houghton crew issues up there?
I know we talk every year.
It's a little informal. It's informal. We do have folks that are advisors to our program. That, you know, I think what was the one gentleman that gave us photos before you guys went up the first time?
Dr. Zacny.
Yeah, Dr. Zacny. So it's informal, but yeah.
Yeah, when you are out in such a remote location, everybody is your friend. You have to take care of each other when you're out there. It's resources are very slim.
Any other questions? Okay. Oh, go ahead.
Is this designed to be a perpetual Groundhog Day station, or is it planned for growth?
We have some very exciting plans for growth. I can talk a little bit more about after for engineering plans on, I believe one of the original ideas was that we would do an overwinter. So the structure is sound, the systems are what we need to make them very robust to survive. That is an enormous challenge.
So if you'd like to help us fund that, you can do that.
That's the first challenge.
Okay, awesome. Well, thank you guys. Thank you so much. All righty, our next speaker is Dean Bergman from Honeybee Robotics. And he's coming to the stage now. Do you have a microphone? Oh. We're going to mic him up. So Dean works with Kris Zacny. Kris has been a presenter at our conference multiple years in a row. It's always a popular presentation. And Honeybee now has been acquired by Blue Origin. And so just love to hear the synergy that's been going on lately and how we're going to get some of these systems up and running. And then after this, we have Dr. Bhavya Lal, who will be presenting virtually. I just talked to her. She's ready to go. She'll be talking about space nuclear power.
Is Dr. Verma's presentation at a different time, later or tomorrow?
Unfortunately, she had to cancel. You can actually access previous presentations of Dr. Vandy Verma at our conference on our YouTube channel.
Great.
All right, Dean,
Thank you.
take it away.
How do I change? Do I just change on the?
Yeah, just use there.
Great. Hi, everybody. I'm Dean Bergman, Director of Business Development at Honeybee Robotics. Apologies that Kris couldn't make it. I'm a poor substitute, but I'll do my best. He got the call up from the big boss, so unfortunately had to be on that call. I'm sure you can understand. So just a bit of background. I think a lot of you know Kris better than you know me. But I originally from South Africa, and that's a weird accent. Got my PhD actually right here at USC in aerospace engineering. Then worked at NASA Ames for a long time on the icebreaker mission, and then resource prospector. So I'm going to talk about a couple of things today, but I thought I'd give you an overview of Honeybee. I don't know how many of you know a lot about Honeybee, but if anything else, if nothing else, this is just a short update. So we were founded in 1983 in New York City, so we have been around. Got into the space industry in the early 2000s with the Mars exploration rovers. Bought out by Blue in 2022. Started off as kind of a hands-off preferred supplier type arrangements. More recently, we're much more integrated. So you'll see if you email us, we now have Blue Origin email addresses, but we still are Honeybee. Can treat us like Honeybee, and we can still use the Honeybee name. Pretty well vertically integrated between our Colorado division and our Altadena division up the road here. About 400 people through both locations. We've sent a lot to space. So a lot of that is on the motion control side. That's our Colorado division. They build more building blocks of spacecraft, so solar array drives, gimbals, actuators, that sort of thing. Whereas here in Altadena, we're exploration systems, and we build full robotic systems. So well known for our drills and sample handling, but we really do everything these days, from mobility systems that we're sending to the moon, to offloaders, and various other pieces of robots that we're sending all over the solar system. Usually start from clean sheet design. We have some things that end up looking similar at the end of the day, but we build more onesie twosies. We're not a production line. So we'll be more bespoke robotic type systems. And we do kind of everything in-house, from mechanism, structure, avionics, software. We do our own operations. We're actually operating systems on Mars currently. And then we work at the full range of project sizes. So small businesses, happy to work with you, as well as the larger NASA projects. So just how we fit into Blue. I know a lot of people are a little confused about this. So Blue is broken out into business units based on products. So engines. New Shepard is the suborbital rocket. New Glenn is the big orbital rocket, should launch again soon. Lunar permanents are all the lunar landers, so that's Mark 1, Mark 2 crew, and Mark 2 cargo. And then in-space systems split into 3. So the first is Blue Ring, which is big, satellite that you can kind of use for a bunch of different things, hosts different payloads. You can take them anywhere between Venus and Mars. And then the two Honeybees for exploration systems and motion control. The three subdivisions of in-space systems, all roughly the same size, although that's diverging a little bit now. So we are located up the road, a mile from JPL. We've got two buildings, a 30,000 square foot lab, which used to be a Walmart that we now turned into a robotics center, and then 30,000 square feet of office space. We were mildly affected by the fire. We were on the fire line. So if you look at where we are, Lincoln and Figueroa was the corner of the fire, and that is exactly where we are located. So we are south of Figueroa and east of Lincoln on that corner. So there were embers on our roof, houses burnt down just north of us and across the wall to the east, but luckily the building was spared. So cleaned it up over a month, a heroic effort from everyone involved. But we are back there working hard. Then inside, we do all of our plant protection and contamination control, which is going to be a bigger focus of this talk. And then we have multiple vacuum chambers, clean rooms. We've built some of the cleanest pieces of space hardware for Mars, so we know how to do that. It's really tough, but we'll talk a little bit about that now too. And then we do avionics labs, geotech labs. And if you're not from LA and you're flying in, come to visit us, flying to Burbank. LAX is very far away. These are some of our, I'm not going to go through all of these now, I'd rather get onto the Mars stuff. But these are some of the missions we're currently working on. So we're going all over the place. Only mission to Mars currently is to Phobos, which could be a pseudo-Mars sample return mission, if you believe that there's Mars sample on Phobos. And that's with JAXA. With MMX, we were part of Mars sample return, which I will talk, I'll sort of flash up a slide of what we were doing, but obviously that is unfortunately not happening right now. So this is our current hardware on Mars, before I get to our future stuff. So the rock abrasion tool was actually the first piece of space hardware that we did, that was with Steve Squires. So both of them worked up into the end of life of both rovers. Little grinders at the end of the arm that would grind the top surface of rock and then flip around, and the alpha ray spectrometer was on the back to image them. Then we did the scoop on Phoenix. So the rat was the first man-made device to grind rock on another planet. The scoop was the first man-made device to touch ice on another planet. So we take a lot of pride in that. The sample carousel inside of the SAM instrument on Curiosity is still operational, and we operate that from Goddard. We have people on site at Goddard that operate it. And then we also had a dust removal tool. It's kind of like a spinny brush that JPL operates. And then for Perseverance, we have three pieces. So we have the witness plate assemblies, which is this is debatably the cleanest piece of space hardware ever built. It's highly polished gold, and that we put inside of the sample tubes. Every now and then, Perseverance takes one out, punctures it, waves it around, and then those will hopefully come back to Earth with the rock cores, and they'll be used to look at the contamination coming from the rover versus what's actually from Mars in the rock cores. We also had a dust mitigation tool with kind of a little sleeve with flexures to clean off the sample tubes, as well as the way that the Perseverance drill does rock coring is actually Honeybee IP. We did not do the drill, JPL did the drill, but they used some Honeybee IP for that based on an SBIR that we did with them. Okay, so getting on to the main part of my talk. So we do a lot of testing in the field, because often what we find is that lab just won't do. And, a lot of what, Mars Society does is the same by putting people in the field. It's much better than doing it in a less more manufactured environment. So we take our hardware all over the world. We try and find locations that mimic Mars as much as possible. So dry valleys in Antarctica. We do a lot of search for life missions. So dry valleys, the Atacama Desert, those are the big two that we go to. But then also we go to the Arctic. We also go to Devon Island with the other Mars guys to do some drill testing there. We also have been to Rio Tinto, Spain, and a bunch of other locations. So these are, we also take, some of our other drills all over the place. Just some cool pictures. This one was a Mars, that one and that one was Mars relevant. Some of the deeper drills are Mars adjacent. You can think of some deep drilling stuff, but I'll show you some other deep drills that we're developing for Mars too, as we get through this. So one of the bigger campaigns that we did was the ARIDS mission with NASA Ames. PI was Brian Glass. I was actually doing this for my postdoc while I was at NASA Ames. It was a big part of my postdoc. So what it was a mock search for life mission. So Icebreaker was a discovery class mission that was supposed to hopefully go to the poles of Mars, drill into the ice sheets, and look for signs of life. So we kind of took that methodology and those instruments and put them on one of Ames's rovers and drove them around the Atacama Desert. So Atacama, one of the driest places on Earth, similar to dry valleys, just slightly easier to get to, and lowest concentration of life. So the idea is, if you can't find life there, you're absolutely not going to find life on Mars. So one of the big, the main goal of this was to practice, autonomous operations. When you're drilling into ice and you hit a fault, you can't stop drilling, otherwise you freeze in place, and not enough power to break yourself free. So it was a big focus on autonomy and remote operations, as well as the testing out the science instruments' ability to search extremely low concentrations of life. But in doing that, what became a major focus was the cleanliness protocols. So obviously there's a lot of humans around. So we had to go through, various stages of cleaning the drill, keeping it clean, wearing bunny suits, as you can see. That is me in a bunny suit on the image on the far right. And, between drills, cleaning it. And we did various tests where we would clean the drill before we started, and then just drill and let contamination build up. And then we would do other holes where we would clean the drill, bring it up, and then clean it between drills. And then we would compare those samples to each other. There's some papers written on that by Arwen Dave. I highly recommend you go look those up. But just thought you'd like some cool pictures of the Mars-like instrument in the field. And yes, I did spend a lot of time sitting next to the drill, making sure that nothing went wrong. So in seeing how difficult it was to keep stuff clean, part of what we do at Honeybee is try to mitigate these. So I want to show you some technologies that we're developing that will help this. So, current state of the art is clean everything before you send it, and then keep it clean through launch and get it to Mars clean. That is pretty tricky because, obviously we'll clean the drill, clean the robotic arm, clean all the things touching sample, but is that good enough, right? Your feet, your landing pads are touching the Martian surface. So when you're searching for life, and there's such low concentrations, and humans are extremely dirty. We put, bacteria on everything. So is it good enough to, just clean before you go? And we say probably not. So we are developing systems to clean in situ and clean in flight. So we've been developing a system called SHIELD. The PI on this is Emily Seto. She runs our Planning to Protection and Contamination Control group at Honeybee. What it is, it's a system that uses UV, pneumatics, and heat to clean either during the flight or in situ. This one was kind of for a Mars sample return type thing, but the shape is also kind of made for drills. We know a lot about drilling, so most of our things are drill shaped. So you could have it attached to the bottom of your drill, go down, drill a hole, and then clean it on your way up or on your way down again. So, you know, a lot of focus on this one. We have Planning Protection Research grants. We have about 3 of them focusing on this. And we just got a Matisse to also focus on developing this. So hopefully be at TRL 6 within the next 3 years based on the Matisse. It would be remiss if I didn't talk about the other things we're doing. So, you know, yes, keeping things clean is a major part of this, but in terms of search for life, where should we go? What type of mission should we do? So this is kind of a mission concept that we've been putting together. I should have swapped the slides around to in Redwater first, but we developed a drill called Redwater. That's this one over here. It's for collecting water on Mars. I'll run through it next. But we have another concept of it where it's on a lander, but instead of melting water and collecting the water, we rather put instruments at the bottom inside the drill, and we take the instruments down hole rather than going, you know, tens to hundreds of meters and having to bring all of that sample back. That would be really difficult. So we're looking at putting instruments inside of drills and taking it down. Also helps with the cleanliness. Then we don't have to, you know, worry about all of the contamination on the surface. Once we get low enough, we can be fairly certain it's clean enough. So this is not just a concept, we are actually working on this. We are working on a project with Jen Heldman, which we call, our part of it is called Smart Drill. So this is putting instruments like spectrometers, neutron specs, IR cameras, all sorts of things inside a drill. So done a wide range. We've got various drills that we do this with, a bunch of different grants that we're developing these technologies. Then Redwater is the piece of it that is actually at TRL 6. So it was, let me see if we can get the video. There you go. So the idea is you drill down to the ice sheets or ice wells on Mars. Then you melt the ice and then use that melt pool to melt more and use a rod well system to create a big cavity. And then you suck up all of that water for human use or science use or kind of whatever you want it for. So I just wanted to talk about Trident a little bit. So some of you may have seen it on the moon recently or on moon missions. This actually started off as a Mars drill. So I wanted to bring it up here to just remind everyone that this was a Mars drill. And, you know, it is built and flown now. So when we're ready to send drills to Mars, this is ready to go. Right now, it's on the moon as part of the Prime 1 mission with Intuitive Machines, and it will be flying again on the Viper mission to the poles. So this is it on the moon. Unfortunately, the lander is on its side, but we got a cool picture of it pointing at Earth. We were able to run through all of the operations. So we, all of the actuators, all of the launch blocks released, and we could run the temperature sensor inside of the drill bit. So we drilled the exosphere of the moon. Unfortunately, did not drill into the moon, but it is done. There, oops, let's see, is that going? So I should talk a little bit about Mars sample return. We were doing the spin eject mechanism. That's the last piece that needed to work for Mars sample return to get the samples back. So that's this ring at the bottom of the sample return capsule. So after, you know, Mars sample return, Perseverance collects rock cores, puts it in a another vehicle that launches the cores into space, and a satellite comes and collects it, cleans it, puts it into the sample return capsule, then all of that comes back to Earth, stops 3 times the distance of the moon, and then we shoot it off with the spin eject mechanism, and it re-enters Earth and lands ballistically, passively and ballistically into the Utah test site. So no parachutes or anything, everything is designed to crash land, because then you can be sure that you're getting your samples back. So we were the last thing, highly accurate device, really difficult to do. We have the prototype sitting in our shop for hopefully my sample return to come back one day, or humans just to bring it back. So either way. But that's about it. So happy to take questions. Also happy to stay during lunchtime and take questions.
While it would appear unlikely that any sample brought back from Mars has native Martian organisms in it, if there were, they would be 100 times more valuable scientifically than all the rest of the sample.
Agree.
So are these people actually planning on preemptively sterilizing the sample? We spend $10 billion to get the sample, and they would destroy the main scientific evidence in it?
So there's a big effort being put in to break the chain. The big reason that MSR was so expensive was because of break the chain. So, you know, when people started doing these studies for, you know, how could you do MSR cheaper, you know, one of the ideas was, why can't we just bring it to Gateway and have people bring it back? But that becomes all of this difficulty with, you know, getting human spores in these samples, because if there is life, we wanted to be able to see it. So these are really clean devices, and everything on the sample, on the chain to get the samples back is done to keep them clean and preserve them.
Yeah, but question again, are they planning on sterilizing the sample brought back from
I
Mars?
don't believe so. I'm probably not the right one to ask about that. You probably need someone from JSC.
So why are they funding you to build a sample sterilizer?
That's to sterilize the drill, not to sterilize the sample. Because if we put our contamination on the drill, we don't want to see that in the sample. We want to see what's in the sample.
Okay.
Yeah, no, we absolutely will not sterilize the sample. That was a good question. Sorry for the confusion. Yeah, the mic is on its way. Sorry. He's going around.
So another question about the sterilization thing on the drill. So you mentioned that it would also be used to sterilize the drill in between digs to make sure that every sample was not cross-contaminated?
Yeah, the idea with that is, you know, not, so the first one is obviously we don't want our sample in the sample, but the idea of sterilizing beyond that is if there's something in the top sample, you want some sort of stratigraphy.
Right. So my question is, how are you able to be sure that that's the stuff on Mars is being sterilized correctly? Because you said that it can be similar to stuff on Earth, but there are some differences in Martian regoliths. So how will you be able to test and make sure that the drill is being cleaned and sterilized properly of all the Martian stuff?
Good question. We have a decent understanding of what could be there. And also these methods that are being used are, you know, state-of-the-art methods for cleaning currently. So sure, there could be stuff that we don't know about, unknown unknowns, but there's not a whole lot we can do about that. So that's the idea of UV and heat and pneumatics. You know, then you can be fairly sure, and you know, we'll test the system to, you know, get rid of as much as possible, and then characterize beyond that if any is remaining. Sorry.
Just curious, are we using chemicals or are we using something like micro lasers for our sterilization process?
So when we were in that Atacama Desert, we were using chemicals, but for Shield, we don't put any chemicals on it. That's all heat, UV, and pneumatics. I like the cap. I miss Maston.
Thank you. Would you mind going back one slide?
Yeah, of course.
So you mentioned this was passive and ballistic.
Yeah.
So this one activity that you see here is the initiation, and it's just going to go all the way.
Yep.
That's amazing.
Yeah, it's extremely accurate. That was the problem with this, because it's like, oh, spin eject mechanisms have been done on various missions. Why is this one so difficult? The accuracy and the fact that it needs this big center hole made it really difficult to do. And there were a couple of companies that bid on it, but there were only 2 viable options, and we ended up winning on the technical. Just, we're not an ejection company, right? But we just understand the technology well enough that we could design something novel. We're good at designing new things, and that's why we were selected to do it.
I like seeing your pore drill. I did a sample return concept in 2008, and there was a polar variation of it. And the sample return for that one was you'd have a disk every day that you'd swap out, and then you just stack the disks up so that you'd know which strata of the ice you'd gone through to go through. Is there anything similar to that in that concept, or is it purely a water recovery system?
So Redwater was purely a water system. Impact is kind of all in situ stuff. So I did not show any Mars sample return concepts for good reason.
Even local analysis.
Yeah, so local analysis, we like the idea of taking the instruments down to the instruments downhole to the sample, so you get the stratigraphy that way. For the ice, you wouldn't get any stratigraphy necessarily. We've been kind of looking at ways of doing ice transport, because we are looking at missions of collecting the ice for science reasons, rather than Redwater melting it and collecting it more for human and ISRU reasons. So there's ideas. There's nothing like the disk thing. That's a kind of a cool idea. Look into that.
Wasn't Honeybee involved in the Planet Vac and Lister instruments that flew on Blue Ghost? Do those instruments have utility on the surface of Mars as well?
Yes-ish. So yeah, thank you for bringing them up. Yes, we did have 2 other devices that just flew to the moon. So Planet Vac is a pneumatic sampler that collects sample and collects it on the surface and delivers it to a sample container. And Lister is a pneumatic drill. For Planet Vac, or for both of them, pneumatics work better in vacuum because pressure to vacuum, highly explosive, so it works really well. You could pressurize it enough to work. We actually have a concept for collecting sample from a drill on Mars using a Planet Vac-like system. The problem with something like Lister is on the moon, we kind of expect smaller rocks that we can kind of push out the way with the pneumatics. Mars, less so. We kind of know that there's probably big things in the ground that are going to be difficult to get through. So there's some application for it, but not as much as you really just want to get a drill down there so you can cut through some stuff. Yeah. Well, thank you.
Thank you very much. Dr. Lal, can you hear me? You can go ahead and turn your video on now.
Hi there. Yes, I can hear you. Can you hear me?
1 second. Let me see if I can increase the audio here. Go ahead and say something again, Dr. Lal. Sound
Hi,
check.
do you hear me? Can you hear me?
Understand.
How about
Is there a
now?
way for you to boost on your end the sound a little bit? We can hear you, but you're a little low.
Okay, I have it at high. How about now?
I think we can make that work, yeah. So let me go ahead and introduce you.
Maybe
Thank
I
you so
should
much for
try
joining
phone
us today.
video?
I'm sorry, go ahead.
Do you want me to try my phone video?
Let's see. It does sound kind of low, doesn't it?
Okay, why don't I speak and you tell me if it's working. Is it working? Can you hear me?
Seems like it's on. Stand by, we're going to troubleshoot a little bit on our end here.
Okay.
Go ahead and say something else.
How is now? Is it better?
Kind of short.
How is now? Is it better?
Yes, I think we can work with that.
Okay, excellent.
I'm going to go ahead and introduce you now.
Wonderful.
So Bhavya Lal is a professor of space policy at the Rand School of Public Policy. Most recently, she served as NASA's Associate Administrator for Technology, Policy, and Strategy, advising the NASA Administrator and senior leadership on a broad spectrum of issues. She was the first woman in NASA's history to be named Acting Chief Technologist. In the first 100 days of the Biden administration, she was the highest-ranking administration representative at NASA, also serving as its acting chief of staff. At NASA, she was instrumental in advancing the Moon to Mars Artemis program and spearheading initiatives in planetary defense, space sustainability, quantum science and engineering, artificial intelligence, space ethics, and space nuclear power, which is the subject of her talk today. Dr. Law, go ahead. We'd love to hear what you have to say.
Thank you so much, James. And yes, I'm a nuclear engineer by training, and nuclear is my jam, as you can tell from my earrings. And I don't know if you can see, but I'm wearing my Perseverance rover necklace, and there's a little Ingenuity helicopter over here. And as you know, Perseverance had an RPS, a radioisotope power system. But we are going to be talking today about fission power. And the premise of my talk is solar lets you visit, nuclear lets you build, and the case for nuclear power on Mars. James, is my sound okay? Am I good with that?
Yes.
Outstanding. Good almost afternoon. Good morning. It's a pleasure to be here with people who understand that power is not a side issue in exploration. It is the issue. Without it, we cannot survive, we cannot build, we cannot stay. And as we look toward Mars, the lesson is clear. Solar lets you visit, nuclear lets you build. Today, in the next 15 minutes, I want to make the case that without nuclear power, human presence on Mars will always be temporary. Let me start with a simple observation. Every great leap in exploration, from seafaring to aviation to computing, began with a shift in how we produced and used energy. And space will be no different. When we talk about Mars, we often focus on propulsion, launch windows, and trajectories, and really important things. But to me, the deeper question is, what happens when we get there? What will power us once we get there? And so that's the case for nuclear power on Mars. Mars is a tough neighborhood, you know that. Solar power drops by more than half compared to Earth, about 40 to 50% of Earth's solar flux at the surface. Dust storms can blanket the planet for weeks. Opportunity rover, one of NASA's and my most beloved missions, died because its solar panels couldn't breathe through the dust. Now imagine sustaining human life, operating habitats, producing fuel, and running ISRU, in situ resource utilization, all through these storms. You cannot launch sunlight into Martian night. You cannot power a settlement through a planet-wide blackout. According to calculations done by my friend and colleague Roger Myers, without nuclear power, it would take roughly 14 football fields of solar arrays to meet the power demands for a crewed Mars return mission. And that's before accounting for dust losses or storage inefficiencies. Nuclear power changes that equation. It offers continuous, high-quality electricity anywhere, night or day, storm or calm. It's what turns a temporary camp into a permanent base. In terms of the amount of power needed, when we model what's required for even a minimal Mars outpost, life support, mobility, oxygen extraction from regolith, you're looking at tens of kilowatts just to survive, hundreds to build, and eventually megawatts to thrive. That's a scale where nuclear shines. A 100-kilowatt fission system, roughly the output of the International Space Station's entire solar array, can support continuous outpost operations through night and storms. It can power labs, refine oxygen, charge rovers, and enable construction. It's a difference between sorties and settlement. So the question is, why now? We've been here before, conceptually, obviously not operationally. For decades, space nuclear power lived in the realm of almosts, promising studies with no missions, funding without deadlines, and leadership without authority. That is now changing. For the first time, all the pieces are aligning. NASA has officially, in writing, selected fission power as a primary energy source for future Mars surface missions. Let me actually really emphasize that. In the history of NASA, that has never happened. There has been no actual specific mission pull for nuclear power or propulsion by NASA or any other government agencies. In fact, NASA has gone out of its way to say that they are not ready for nuclear propulsion yet. They're still working through all four alternatives: all chemical, NEP, nuclear electric propulsion chemical, NTP, nuclear thermal propulsion, and SEP, solar electric propulsion chemical. NASA has not made a down select. However, on power, NASA has made a down select, and that is unprecedented. This is not something we are seeing on the DoD, Department of Defense side either. DoD has made no decision on whether for any of their missions they would like to see nuclear power or propulsion. So this is a very unique moment for us. And the moon is our proving that ground. A tens to hundreds of kilowatt class fission surface power system by 2030, which is what NASA has recently announced, if you're paying attention, will demonstrate everything we need for Mars: autonomous operation, dust-tolerant radiators, regolith shielding if needed, and a closed power loop. Fuel is finally available. Department of Energy has produced the first significant domestic supply of what's called high-assay, low-enriched uranium, about 900 kilograms by mid-2025, enough to support early reactors in both space and on Earth. And for the first time ever, we have policy clarity. The United States has a national strategy for space nuclear power and propulsion through SPD 6, Space Policy Directive 6, that came out during the first Trump administration. And there is also a clear launch approval process, which is called National Security Presidential Memorandum 20, NSPM 20. So we now have a pathway, a need, and a timeline. But the challenge, as always, is execution. Even with all this progress, history tells us that having a plan isn't enough. In fact, execution is where we always fail. Our problem has never been physics. It has been follow through. We start too big with no precursor missions. A really good example was the JIMO Prometheus mission from the early 2000s, where we planned to go to the moons of Jupiter, big 400 kilowatt electric system, NEP system, no precursors. We launch before we test. In fact, the Greco program, the DARPA NASA collaboration to have a nuclear thermal propulsion mission, planned to, did not plan a ground-based test. It planned a test in space. We chase propulsion without a power base. NTP in particular is hard enough. We need reactors operating at 3000 Kelvin. We need to get that reactor piece, settled before we add the propulsion piece. We designed for ambition without a customer. Up until NASA in November of 2024 put the stake in the ground about needing power on the moon, power on Mars. There wasn't ever a customer. So all the previous times we started to develop nuclear systems, we were doing it as a tech push, not a mission pull or a demand pull. And that makes all the difference. That's why our report, one I wrote with Roger Myers, it's called Weighing the Future, Strategic Options for U.S. Space Nuclear Leadership, and you can easily find it online, argued for a power-first strategy because it's an easier first step. Our report lays out 3 options for space nuclear leadership and provides a roadmap for how we can finally move from paper reactors to flight hardware. Don't get me wrong, propulsion is essential, but it builds on power. Both power systems and propulsion systems need reactors, as I just said. Both depend on the same fuel, similar fuel, maybe not the same fuel, since, as I said, NTP requires a reactor operating at much hotter temperatures, similar infrastructure, and regulatory frameworks. If we start with propulsion, we still have to build everything power requires, just without the near-term payoff. But if we start with power, we actually accelerate, we just de-risk propulsion. So NASA's decision maker in announcing the Fission Surface Power Program got it right. Power first, propulsion next. Build the backbone, then the engines. In our report, we actually talk about something we call the Manhattan Project test. So the idea there is it isn't enough just to announce a grand program. What you need is you need to meet 3 conditions, and that's our Manhattan Project test. You need a single empowered lead with real budget and milestone authority. We haven't really done that before. We need stable, multi-year funding insulated from annual swings. Something will go wrong, as it always does. This is hard to develop. Shutting down at the first sign of trouble is not a good idea, and there will be trouble. This is hard. Third, and this is what this is the hard part. We need a strategic imperative that's strong enough to align institutions such as, NASA with DOE, with DOD, with NRC and other government agencies that are relevant. So you need a strategic imperative that's sufficiently strong that all these institutions can stay aligned and sustain momentum when the going gets tough, when administrations change. One reason GMO Prometheus got canceled, as many of you know, is the new administration came, the shuttle had to go back in service. There was a $1 billion shortfall. GMO Prometheus wasn't doing well. That's where the money came from, right? So we have to learn from the past. We have to learn from the past mistakes as well. Miss any one of these and you end up with another GMO Prometheus. Remember that. We spent hundreds of millions of dollars in that program, but little to show because we overreached without a strategic imperative. And obviously, when push came to shove, the program got canceled. If we can meet these 3 conditions, we can fly to the moon first by 2030 and then to Mars. If not, we risk slipping into the familiar pattern of delay and decay. Now, let's talk about governance for a quick minute. Today, we have a clear launch authorization framework. I mentioned NSPM 20, but we don't yet have a settled process for operating a reactor on another world. That's the next frontier, what we call mission authorization under Article 6 of the Outer Space Treaty. So for those who are less familiar, these are the rules that govern how nations launch and operate things in space. And they are what give the public confidence that this can be done safely. So we need to define how safety zones around these nuclear power plants are set, how systems are going to be supervised, how they will be decommissioned. We also need to settle what's called indemnification, who is legally responsible if something goes wrong. On Earth, the Price-Anderson Act provides the clarity to commercial nuclear power plant providers. In space, we have none of this. So these aren't showstoppers, but they're execution problems. And the fix is clear. embed regulators from day one, integrate safety reviews into program milestones, and put licensing on a clock. Last, I want to talk about the new geopolitical imperative. Because whoever fields the first continuous reactor in space, on the moon, and then Mars, will shape the norms for everyone else. A continuously operating system defines safety zones, operating procedures, and how things like harmful interference can be interpreted under the Outer Space Treaty. If China lands first and declares a multi-kilometer safety perimeter at the lunar South Pole or on the Martian ice fields, that becomes the precedent. Functional control without formal claims. Our answer, of course, should be transparency and leadership. Define hazard-based safety zones, publish the technical rationale, invite allies to align. If others follow, we set the norm. If they don't, at least the rule book is written by us, not imposed upon us. And what does success look like? It isn't just flying one reactor. Success is building a sustainable space nuclear ecosystem that endures beyond any single demo. By the mid-2030s, I want NASA and commercial users to buy power in space and on the moon by the kilowatt hour, just like on Earth. By the 2040s, I'd like to see fleets of reactors serving civil, commercial, and defense missions, with US standards becoming the global default. That's what lasting advantage looks like. Systems others depend on, rules we define, and services that are too valuable to ignore. So let me just end, if I think that I have about 15 minutes, by saying that I often say that my goal in life is to bring the solar system into our economic sphere. Nuclear power is how we start. It's what lets us end poverty in space, on the moon, on Mars, and beyond. It's what transforms exploration into infrastructure, sorties into settlements, visits into presence. If we get this right, we will not just reach Mars, we will stay, we will build, we will thrive. Let me end by saying what I started with: solar lets you visit, nuclear lets you build, and building is how we lead. Thank you.
Thank you, Dr. Law. We have time for a few questions. Here's Robert Zubrin.
Hi. So could you just tell us what is the reactor that you intend to develop? What is its power? What is its power conversion? And how much is your budget?
Okay, so I'm not NASA. I cannot speak for NASA, but I can tell you what is available in the public domain. There was a draft RFP that was released by NASA. And before the draft RFP, the interim administrator, Sean Duffy, had put out a memo. And the memo essentially talked about 100 kilowatt electric, 100 kilowatt plus electric system using a Brayton power conversion system, which actually was an interesting decision since it has, Brayton has extensibility. So, as the reactors get bigger, you don't need to switch from Brayton to another system. So that's a conversion system. The requirement was to keep the system under 15 metric tons. Basically, that is something like a Starship launching it, landing it. Although in principle, Blue Origin's Mark II lander can also. And again, I'm not 100% sure about that. With respect to budget, I don't think that NASA has set any plans, but in the Duffy memo, they talked about using essentially the line item, the president's budget request that PBR had, which was about $1 billion, and it had numbers like 350 million in the first year, 500 million in the second year, and then on from there. But best I know as an outsider, I'm not NASA again, best I can tell, these funding levels haven't been set yet. Did I answer your question, Bob? Did I get all of the sub pieces you had?
Yes, it does. Thank you.
Cool.
Hi, Dr. Law. Thanks for joining us. I know a lot of people, when they think about the economics of space exploration and the benefits from technology development and all of that, they really like the idea of, space technology spin-offs. Given that fission power is already a proven technology that we know and understand and have markets for here on Earth, I'm curious if you think that there are additional benefits, if there's more meat on the bone from the further research and development into space rating these reactors and also, ramping up stuff with propulsion that could positively impact terrestrial markets.
Okay, great. The question is sort of what are the synergies between space systems and terrestrial systems? So right now, terrestrial nuclear power plants don't operate at very high temperatures for safety reasons. And as you know from thermodynamics undergraduate courses, the higher the temperature, the higher the efficiency. So coal power plants on Earth today, in fact, have higher efficiencies than nuclear power plants because coal power plants operate at higher temperatures than nuclear ones. So, one way terrestrial nuclear power plants can be better or have higher efficiencies is if they could operate at higher temperatures. Put a pin in that. Space reactors, for reasons because they're in space, have to operate at higher temperatures. The reason for that is there is no convection or conduction in space, or not much. Most of the heat rejection is through radiators. And from the Stefan Boltzmann law, sorry, I'm referencing a lot of undergraduate thermodynamics here. The higher the temperature, the lighter, the smaller the radiator will be. So space reactors tend to, or at least in design, need to operate at about 1,000 degrees. And now we have to have fuel and materials for these higher temperature reactors. Once we develop those, voila, we now have the ability to have higher temperature reactors on Earth. And now Earth nuclear power plants have a higher efficiency. So that's one really big advantage and a piece of synergy between Earth and space reactors, where developments in space directly benefit developments on Earth. A second one is high assay, low enriched uranium. So up until now, our reactors in space, and by the way, if you all know, we have launched a fission reactor in space in 1965. And up until now, we only use highly enriched uranium in space. And the new ideas are that we can actually potentially operate space nuclear reactors with what are called high assay, low enriched uranium HALEU fuel. HALEU fuel is enriched up to 20% as distinct from HEU, which is 93% and higher for that's what's called bomb grade enrichment. Demand pull for HALEU reactors in space, basically adds to the demand pull for HALEU reactors on Earth. A lot of the next generation reactors would prefer to operate with HALEU systems. Just FYI, for those of you who don't know, current reactors, previous generation reactors are at about 4 to 5% enrichment. So space systems, which need 20%, now it's 19.75, but basically 20% enrichment will then help Earth reactors also get the fuel that is 20% enrichment. So those are two things that come to mind. On your issue on propulsion, for propulsion, NEP, nuclear electric propulsion, is essentially a power reactor with thrusters bolted on. Thruster technology is, we have Gateway, for example, has thrusters, I think 12 kilowatt thrusters, and again, other companies have developed thrusters in the tens of kilowatts. I'm not sure of Earth implications for NEP or NTP. NTP is a different design altogether. NTP reactors are more like chemical propulsion systems where you separate out the heat, the heat source from the propellant, right? So in a chemical reactor, some oxidizer and the fuel, they mix in the combustion chamber, and gas comes out the nozzle at high temperature and high velocities, creating the Newton's third law, the push to move forward in a nuclear thermal NTP system, nuclear thermal power, nuclear thermal propulsion. The nuclear reactor is a heat source, and now you can actually have any propellant you want. You are not limited to having, basically H2O, and hydrogen is the best one. It's the lightest molecular weight, which means you can get the most specific impulse out of it. I am not sure of any synergies with ground, other than, again, we are developing high temperature reactors, which will help terrestrial systems. But I think if you were talking about synergies between power and propulsion, there's huge amounts of synergies between power and EP, nuclear electric propulsion. A little bit less so between between power and NTP, although obviously there's a huge amount of development insights you get by just developing and launching nuclear power systems, because once there's existence proof that you can power, you can have a power reactor in space. Now you're just developing higher temperature systems. I hope I answered your question.
Hi, Bhavya. It's Matt Kaplan. Are you familiar with the status of China or any other nation's work development for either power or propulsion?
No more than what's in the media. There's articles that China is going to be collaborating with Russia to develop a megawatt level nuclear power, so a fission power system for the surface of the moon. I have tried to, but not been able to track down this, memorandum of understanding. But that is what we know. So that's China. Again, they have plans for, papers that have been written on both power and propulsion, and writing papers is easy, as we all know. Doing research is easy. We don't know where they're at with respect to, for example, test facilities for testing an NTP system, which will likely be very, very expensive, because you don't want radioactive fission products going out the back in test facility. On other countries, the United Kingdom and France are looking pretty carefully at new source material for radioisotope systems. So using americium instead of plutonium-238, which is what the United States uses. So that's an interesting opportunity for collaboration. And also, there are companies in the United Kingdom that are developing engines that could be relevant to nuclear thermal propulsion. That's what I know of. And again, if there's folks who know more, I would love to hear from you.
Are you aware of any efforts by NASA or the DOE to evaluate the suitability of some of the microreactor designs that are being implemented by a lot of these startups in the United States? Like, for example, Radiant Power Systems that is down in El Segundo?
So that's a very exciting thing about the announcement that came out. And by the way, our report also recommended that approach, that instead of developing these systems in-house, NASA used the commercial sector and write fixed price, milestone-based contracts with these companies. Radiant is one, another one is Antares, Space Nukes. So there's several companies that that are working on terrestrial reactors, and it would be fantastic if they would bid on for this fission surface power program that has been announced. And I'm assuming that larger companies would bid as well, but I really hope that it's a very it's a vibrant group of companies that bids. I hope that NASA is able to pick more than one company. As I said earlier in my speech, I think it is important that there be a competitive power market on the moon, in space, on Mars, and beyond, and that it be commercial. I mean, the terrestrial nuclear power enterprise is entirely commercial, except maybe TVA, Tennessee Valley Authority, which I think may be some kind of a public-private partnership. But most power plants are privately run. Enrichment is privately done. all of that is the entire nuclear supply chain is private on the ground and terrestrially. There is no reason why it ought to be government owned and operated in space. So that's kind of my vision. And I think NASA is kind of on that track. I hope that they get really good proposals and they get to, award more than one awardees.
Hello, Bhavya. My name is Rahul, and I go to high school in New Jersey. And I wanted to talk about your experience with different types of nuclear fusion reactors. For example, I came across magnetic confinement fusion. And in this type of reactor, powerful magnetic fields contain the plasma in the donut-shaped vacuum chamber or the toroidal vacuum chamber. And this minimizes energy loss and basically enables nuclear fusion to happen. And I was wondering how this changed the total mass of the reactor, and would it make it easier and less affordable, or more affordable and less cost to transport reactor to different places like the moon and Mars?
So yeah, I have no idea how to answer your question. It is, yes, I am a nuclear engineer by training, but it was a long time ago when I was doing these calculations. So you'll have to email me separately and I'll have to think about that. And I can also connect you with people who actually do these kind of calculations. The thing I would say is it is very important to study future concepts, right? I mean, use of fusion. And there's some really amazing companies that are doing fusion research, and I'm all in, and I think they should get private funding, they should get government funding, and we need to keep on this R&D curve that each generation is better. But I also want to fly something. I want to fly something in the next 5 years. And the question is, what is it that we have that is ready enough to fly in the next 5 years? And I don't think any fusion systems, and again, I'm perfectly happy to be wrong, but the best I can tell is that no fusion system is ready to fly quite yet. And let's keep working on that. Let's keep doing the R&D, but let's fly the system that we can fly today.
That's an excellent last comment, Dr. Lal. Thank you so much for your presentation today. We really appreciate your time.
It was an honor. I was at the Mars Society in person a couple of years ago. It was just fantastic to be there. It was great meeting Bob and the amazing group that you have there. I still have my t-shirt from a couple of years ago, and I hope you will send me the t-shirt for this year. Thanks, everyone.
We
Bye-bye.
sure will. We sure will. Thank you so much. Take care.
Bye.
Okay, folks, that concludes our morning plenaries. Again, if you want a lunch recommendation, the USC Village is about a 10-minute walk from here. Lots of choices there. We'll be starting back up at 1:30. Just a reminder, the rooms for the tracks. So this room will feature not the first track, but the second one on the schedule, which is legal and political. So the legal and political track will be in this room. The Mars Against Hunger competition finalists will be presenting in 208. Astrobiology track is in 210. And the technology track is in 212. Also a reminder tonight that the banquet is at the Amundsen Senior Center, which is on the USC campus. It's near the stadium. We hope to see you guys there. And if you still need a banquet ticket, you can go downstairs to our registration desk and they can help you purchase that. Have a great lunch and we'll see you this afternoon.
There are no separate banquet tickets. As a video.
Look up with what
All right.
Yeah, that would include. We just want to make sure you will get in. Yes, sir. Okay, good. Of course.
Only one guy.
Yes. If it blows up. Yeah, our sponsor guy. Yeah.
He wants to know. Which is funny because normally we can watch it.
Oh, yeah. Yeah.
Yeah? Okay.
Yeah, we're gonna, so we.
It's just way too much. I can't record each and every.
So actually, like, but like a live element. Like, if I could go get, you know, have a day or two.
So
did you get lunch?
If there's specific things you want, I can get those ready for you by Monday over the weekend. Essentially, the best speeches are in this room in the mornings, and those are recorded. So, like if you're looking at the schedule and there's a